Maricar S. Prudente, also from DLSU. During the time of Dr. Prudente in 2012, the PRF was reorganized and was transformed into JAAP, covering not only the JSPS Ronpac, all the other fellows that had undergone exchange programs in Japan through the JSPS Scholarship and Exchange Programs. A JSPS PIN is provided by JSPS for each JSPS alumni through JAAP. JAAP was registered under the Securities and Exchange Commission in 2013. During the term of Dr. Gaspilio, PRF Connect, which was the official newsletter of the organization, was published. During the term of Dr. Prudente, JAAP started the Research Symposium and General Assembly meeting where JSPS Bangkok, led by Professor Kuniaki Yamashita, started to take part in the activities. Ties with the JSPS Bangkok was strengthened. The JSPS Bridge Fellowship Program was also launched. From 2014 to 2016, Dr. Jaime Montoya, the Executive Director of the DOST Philippine Council of Health Research and Development, or DOST-PCHRD, became the President of JAAP. During his term, the Research Symposium and General Assembly meeting of JAAP became a component of the National Science Technology Week of DOST. At that time, JAAP came under the umbrella of the International Cooperation of DOST, where Dr. Leah J. Buendia was serving as Assistant Secretary. JAAP's official website, www.jsps-jaap.org, went online, and JAAP was featured in the JSPS Quarterly, the international newsletter of JSPS. From 2016 to 2018, Dr. Susan Gallardo of DLSU became JAAP President. During her term, the PRF Connect was revived and rebranded as JAAP Connect. JAAP Connect became the official newsletter of JAAP. The JAAP Outreach Program was launched where Mulanay Quezon was named its first beneficiary. The first JAAP Technology Forum was held at the Central Luzon State University, or CLSU. From 2018 to 2020, Dr. Renato G. Reyes, Vice President for Academic Affairs of CLSU, became JAAP President. His Vice President was Dr. Danilda Hufana Duran, Scientist of the Department of Agriculture Philippine Carabao Center, or PCC. Dr. Reyes launched the JAAP International Research and Development Conference and strengthened the outreach programs in collaboration with the Fulbright Philippines Alumni Association, or FPAAA, where Dr. Danilda Hufana Duran was the president. JAAP also launched in 2019 the selection and awarding of the Outstanding Filipino JSPS Fellows, with ASEC Leah J. Buendia serving as the chairperson of the selection committee. Due to the lockdown caused by the COVID-19 pandemic in 2020, JAAP launched its first ever webinar series in partnership with the International Affairs Office of CLSU. All Filipino scholars and grantees registered as members of JAAP to foster closer ties among them and together realize their full potential for the service of the countries whom they owe their professional excellence.
Central Luzon State University, through the years, has grown into a comprehensive and inclusive higher education institution that transforms the mind and improves the quality of life of peoples and communities it serves. From its humble beginning as an agricultural school, with its vast land resources, CLSU offers a conducive environment for learning, research and production catering the needs of students, faculty, staff, researchers and other stakeholders. For more than 100 years, agriculture has remained the flagship program of CLSU, keeping a multidisciplinary approach through the complementation of the different curricular programs it offers, including arts, social sciences, business management, and other allied fields. CLSU is also recognized as an agri-tourism site in Luzon featuring the different R&D centers that specializes in precision agriculture, livestock, crops and fisheries. These research centers support, strengthen and sustain the thrusts of the university's academic program. CLSU being strategically located at the heart of the Muñoz science community, it has access to national R&D institutions bringing in additional resources, facilities and experts, which further amplifies the research initiatives of the university. CLSU today stands proud as one of the renowned and prestigious universities in the Philippines and Southeast Asia, and is positioning itself in the global community. CLSU, keeping agriculture as a way of life.
The province of Aurora is known as the hidden gem in the east coast of central Luzon. For its lush greeneries, mesmerizing seaside, rich culture, arts, and history situated in one landscape. At the heart of the province lies the future.
Let us unceasingly work together to bring ASCOT to the world, position ASCOT in the world, and make ASCOT relevant to the world. Let our students be global learners, our faculty members and staff as global influencers, our alumni as global ambassadors, our partners as global benefactors.
Being the beacon of standard and quality education for over 26 years, Aurora State College of Technology continues to bring more opportunities for students and faculty. From its humble beginning in 1993 with 301 students and 10 classrooms, the late Senate President Edgardo Angara envisioned an institution that could provide equal opportunity for all. ASCOT has grown and developed into a bigger institution of higher learning, now having 3 campuses. The Sabali campus in Baler, the Basal campus in Maria Aurora, and the Esteves and FTC campus in Casiguran, all fruits of continuous nurturing as seen today. With the full support of the Commission of Higher Education, ASCOT is ready, now more than ever, to be recognized in global spectrum.
Join me in embracing the core values of ASCOT. D for dependability, R for responsiveness, E for empathy, A for adaptability, M for mindfulness.
Welcome to ASCOT, dedicated to empowering minds and shaping the future to the world, in the world, and for the world.
Let us unite to bring out the best of ASCOT.
All right, good morning once again and welcome to another episode of JAAP webinar series of 2025 with the theme Exploring the Diversity and Wonders of Marine Environment, brought to you by the Japan Society for the Promotion of Science Alumni Association of the Philippines in collaboration with the Central Luzon State University. My name is Marjorie Ramirez.
And I am Ace Aguilio, your program host for today.
To formally commence our program, let us all observe respect for the singing of the Philippine National Anthem and the Japan National Anthem, to be followed by a short invocation.
To see we
To officially welcome us to this insightful event, let us hear from our esteemed guest, the Director of Japan Society for the Promotion of Science, Bangkok, Thailand Office, Dr. Yoshio Otani.
Good morning. I'm Yoshio Otani, the Director of JSPS Bangkok Office. On behalf of Japan Society for the Promotion of Science, I'd like to express my sincere gratitude to the continuing efforts of JSPS Alumni Association of Philippines, JAAP, for holding webinar series 2025. The theme of this webinar is exploring the diversity and wonders of marine environment. That title reminds me of the JAAP IRDC 4 held on-site on June 23 to 25 in El Nido with the theme of challenges and opportunities in the post-pandemic era, just after the settlement of corona pandemic. It was my first travel outside of Thailand since I became the director of JSPS Bangkok office. This trip made me decide to continue my position as a director of JSPS Bangkok office. I really thank all the members of JAAP for giving me this opportunity. As an outreach activity of this conference, we visited many remote islands around El Nido, and I was very much impressed by the turquoise blue and emerald green seas. The development of remote islands is an important issue in Philippines, and I think it is a big challenge to develop islands while keeping the beautiful seas. I think the webinar title is very suitable and timely to think the diversity of marine environment and decide the direction of development of the remote island in Philippines. I hope this webinar gives us some insight into how to manage and restore marine environment while developing the islands. Thank you for your attention.
Thank you, Dr. Otani, for those inspiring and thought-provoking words. Your insights have truly set the tone for what promises to be an engaging and enlightening session.
Okay, so before we proceed, let us take a moment to capture this special gathering. May we request all participants to turn on your cameras for a group photo opportunity. Tech team, please take it away.
Good morning. Kindly open your camera and let's take our group photo. Currently, we have 6 slides. So let's start for our first slide. So 1, 2, 3. Next slide. 1, 2, 3. And third slide. 1, 2, 3. Fourth slide. 1, 2, 3. Fifth slide. 1, 2, 3. And for the last slide, 1, 2, 3. Okay. Thank you.
All right. To further inspire us, we have messages from distinguished leaders. First, let us welcome the Vice President for the Academic Affairs at Central Luzon State University, Dr. Ravelina A. Velasco.
Hi, everyone. The JSPS Alumni Association of the Philippines and Central Luzon State University warmly welcomes you all to this webinar on exploring the diversity and wonders of marine environment. Our university president, Professor Dr. Evaristo Abella, sends his warmest greetings to all of you. Also, I'd like to greet Sir President Professor Dr. Renato Reyes, and of course, Dr. Yoshio Otani, and our distinguished speakers, of students, and all the participants from this virtual room. Once again, good morning. Now, this webinar is a fascinating journey beneath the waves to discover the beauty, richness, and importance of marine ecosystem. The ocean is a source of life and driver of climate regulation and a reservoir of incredible biodiversity. From vibrant coral reefs to deep sea creatures to the essential role of the marine ecosystems in sustaining life on Earth. Today's discussion will offer insights, discoveries, and latest trends on research on marine biotechnology and its application, algae, and seagrass. We are honored to have Dr. Kustiaria Tarman, Dr. Waimun Lung, Assistant Professor Marco Flores de Guzman from ASCOT, and of course, our very own Dr. Gary A. Benico. I leave you with this message from Sylvia Earle. And she says, no water, no life. No blue, no green. Once again, good morning, everyone. And let's have a very fruitful discussions on exploring marine environment ecosystem. Thank you very much.
Thank you, Dr. Velasco, for sharing such an insightful and inspiring message with us today. Your words have truly resonated with us. Let's carry these lessons forward and put them into actions in our lives.
So next, let us hear from the President of Aurora State College of Technology, Dr. Renato Reyes. Sir.
Yes, hello, good morning. May I respectfully recognize the Honorable Member of the Board of Trustees of Aurora State College of Technology. I can see here our very own Dr. Julius Cesar Sicat. Despite of his very busy schedule, is joining us in this very important meeting. Our Executive Director of JSPS Bangkok office, Professor Dr. Yoshio Otani, the President of JAAP, Dr. Karl Marx Kiazon, the Vice President for Academic Affairs, Dr. Ravelina Velasco of CLSU, of course, the President of the Central Luzon State University, Dr. Evaristo Abelia, our speakers here, of course, my friend, Dr. Kustiaria Tarman, was my friend and my cohort when we were in Germany. Hello, Dr. Kusti, Dr. Waimun Lum of MIA University, our very own Assistant Professor Marco Flores de Guzman, our professor from Central Luzon State University, an expert in the phycology, Dr. Gary Benico. I also would like to recognize, of course, the Director of the International Affairs Office of CLSU, Dr. Parsons Sahil, and his staff, RJ Marco, Emil, and the rest of the team. A pleasant morning to all of us, to our faculty members, the students who are attending this important gathering of scientists and researchers, our passionate advocates of marine conservation. Welcome to today's deep dive into the incredible world beneath the waves. The ocean is more than just water. It is our source of food, jobs, and life itself. But problems like overfishing, pollution, and climate change are putting our marine resources at risk. If we don't act now, future generations may not enjoy the same rich and thriving seas we have today. You remember that the Philippine rice is located, or the Benham rice, we call it formerly as the Benham rice, is located in the Casiguran area in the east coast of Central Luzon. This webinar, a collaboration with JSPS Alumni Association of the Philippines, Central Luzon State University, and the ASCOT School of Fisheries and Ocean Sciences is not just about learning. It is about finding real solutions. With the help of experts, researchers, and communities, we can take steps to restore and protect our oceans. So, let us ride the wave of change. Let us make protecting our oceans not just a conversation, but a commitment. Because a future with healthy seas means a future where we all win. Thank you and mabuhay.
Thank you, sir, for that inspiring message.
At this point, we are delighted to introduce our distinguished speakers. Please join me in welcoming Dr. Mark Joseph Rafael, the esteemed Dean of the College, the Dean of the School of Fisheries and Ocean Science at Aurora State College of Technology.
Good morning, everyone, and welcome to our webinar series on the wonders and diversity of the marine environment. It is our privilege to have with us today an esteemed panel of experts whose research and contributions have significantly advanced our understanding of marine science. Let us now introduce our distinguished speakers. Our first speaker, Dr. Kusteria Tarman, is an associate professor at IPB University in Indonesia and a leading researcher in marine biotechnology and aquatic products technology, with a PhD in pharmaceutical biology from the University of Greifswald in Germany. She has dedicated her career to exploring marine-derived bioactive compounds, particularly from marine fungi, seaweed, and microalgae. Her pioneering work has broad applications in biotechnology, sustainable aquaculture, and marine-based pharmaceuticals. Dr. Tarman is also deeply involved in the development of marine-based functional foods and nutraceuticals, reinforcing the importance of marine resources for food security. Her global collaborations and expertise in sustainable omega-3 production and bioactive metabolite profiling makes her a vital contributor to today's discussion. Our next speaker, Dr. Wai-Moon Lung, is an esteemed marine biologist and assistant professor at Mie University, Japan, with a PhD in agricultural and life sciences from the University of Tokyo. She specializes in harmful algal blooms and marine phytoplankton diversity. Her research delves into the taxonomy, phylogeny, and ecological impact of marine dinoflagellates, providing critical insights into toxic algal species and their interactions with the marine ecosystem. Dr. Lung's contributions to the field of marine ecology have been widely recognized, and her studies on the environmental factors influencing harmful algal proliferation are crucial for advancing marine conservation efforts. We look forward to her valuable insights today. Our third speaker, Mr. Marco Flores de Guzman, is an assistant professor at Aurora State College of Technology and a dedicated fishery scientist specializing in coastal resources assessment and marine biodiversity. His expertise spans biological and physical oceanography and antimicrobial potential of marine organisms and the ecology of commercially important fish species. His research on fish length-weight relationships and catch composition in Philippine waters contributes significantly to sustainable fisheries management. Additionally, his work on antimicrobial compounds from the marine organisms has promising applications in aquaculture disease management. His dedication to responsible fisheries practices and marine conservation makes him an integral part of today's discussion. Certainly not the least, we have Dr. Gary A. Benico, a leading Filipino phycologist and professor too at Central Luzon State University, holding a PhD in aquatic bioscience from the University of Tokyo. Dr. Benico has made significant contributions to the study of harmful algal blooms and marine microalgae. His research has led to the discovery of 3 new species of marine microalgae and has provided crucial insights into the molecular mechanism of paralytic shellfish toxins. Beyond this research, Dr. Benico is actively engaged in training researchers and fisheries personnel on monitoring and managing harmful algal species. His expertise bridges research and practical applications, ensuring the sustainable management of marine biodiversity. We are honored to have such accomplished speakers with us today, and we are excited to gain insights from the groundbreaking research and extensive experience. Let us give them a warm welcome as we embark on the enlightening discussion on the diversity and wonders of the marine environment. Thank you, everyone.
Thank you, sir. Okay, so everyone is excited, I'm sure. So let us now proceed with the lecture presentation. For our first topic, we are honored to have Dr. Costeria Tarman from the Faculty of Fisheries and Marine Sciences, IPB University. She will be discussing marine biotechnology and its applications. Dr. Tarman, the virtual floor is yours.
Thank you very much. Good morning, everyone. Okay, so first of all, I would like to thank for the organizer to inviting me here, Dr. Karmats, the president of JAAP, and of course, the two presidents of CLSU and ASCOT, Dr. Velasco and also Dr. Reyes. And thank you for again for the invitation, and it's my pleasure to be here to share with you all on the research that we have done here in IPB in Bogor. So thank you for the moderator for the introduction. And besides, as the faculty members of IPB, currently since to 2022, I also as the adjunct professor at the CLSU, and as the alumni of JAAP Germany. At the moment in Indonesia, we have the Alumni Association for Marine Resources Studies. We call it JAMARS. And at the moment, again, I'm as the chairperson of JAMARS. So hopefully in the future, we can collaborate also with JAAP or probably also other alumni of JSPS. So for today's sharing, I would like to share on marine biotechnology and its application. Since I'm at the Faculty of Fisheries and Marine Science, then the focus will be on the use of the marine resources. So for today's talk, we'll begin with introduction and then marine biotechnology and the resources. And the third, related with the present and future research that we have done, and then closing remarks. So, yeah, as mentioned by Dr. Reyes, that we are actually cohort during the training in Germany 20 years ago. So we are here. So we are here in IPB Bogor, and then in Luzon Island for you all, in ASCOT and CLSU. But it's currently the distance is now a problem. So we can see here some pictures that I've been in CLSU last 8 years, and then also Prof. Reyes in IPB, also 8 years ago, and then last year with the delegation of ASCOT. So I think that in the future also more collaboration. Okay, so just short on IPB, the Bogor Agriculture University, probably also before you know about that. So we are located in the south of Jakarta, so it's just like 1.5 hours from Jakarta, the capital city of Indonesia. So we have almost 1,500 staffs and 35,000 of students. So we have 13 faculties and schools, and I belong to the Faculty of Fisheries and Marine Science. So also we have a research institute, and then I'm as the executive secretary of this research institute, especially for maritime, ocean, and fisheries. So we have an IPB 24 of research centers, and these research centers classified into 5 related with the marines, and then climate change, food and health, and then advanced technology and social science and economics. So the research institute, the task is to connect the partners. So IPB with our partners. For instance, here we have from all over the world, including also Dr. Reyes, that we have signed an OU since last year. So when Professor Reyes in IPB in Bogor. Okay, the research focus in our institute related with the marine, maritime and islands state, for instance, and then the coastal marine resources and fishery studies, inland fisheries, and blue carbon and climate change, of course. And from this for focus, so I belong to this part. Currently, also head of the Marine Biotechnology Division in the Center of Coastal and Marine Resources Studies. Related with that, then my research focus on the marine biotechnology. So just to give you a brief introduction on marine biotechnology, is actually the application of several science and technology, including not only related to molecular biotechnology, it's also so. Okay, what happened? Okay, so biotechnology also involves biology, chemistry, genetics, even economics. So with this, all the application of the science, so we can use marine resources as the raw materials, and then we can make these resources into aquaculture, for instance, using the genetic engineering, so we can have the best quality of fish, shrimp, and so on. And then the second, related with the natural products and for the services, for instance, in the remedy of polluted environment, so using the bioremediation. So on this presentation, so I would like to focus on the second part of the marine biotechnology in regards with the natural products development, not only for pharmaceutical use, but it's also including like nutraceuticals and other industrial aspects. Okay, so as we know that we are in the Coral Triangle hotspot of biodiversity in the world, so including here in Indonesia and then Philippines and other countries that we really blessed with these resources. Not only the terrestrial resources that we have huge diversity, but it also including underwater creatures. So therefore, we should focus also not only the terrestrial resources, but it's also including marine resources. However, since the research related with the marine resources is probably a little bit left behind, so we have the diversity in huge number of the resources, but unfortunately, most of them is still less explored. Not only related with the microorganism, but also including the macroorganism. For instance, we are here in Indonesia, still focus on the fish and shrimp. Even actually, we are one of the biggest producer of seaweeds, but still the value added of this is still low. Therefore, with these resources, we can also use, for instance, for drug development and so on. So currently in the market already available some drugs like anti-cancer, antiviral, painkiller, and so on that originally isolated from the marine resources. So even we have 3 minutes huge number of marine resources. Unfortunately, they still the in the context of value added still low. In our group, we have some research related with the marine biodiversity, especially for invertebrates, because of these resources actually from the natural products report, they have prospective marine bioactive compounds that can be developed, for instance, for the pharmaceutical products and for other bioactive related products. Unfortunately, that with these resources, still less observation on that. So that's why in our group, we try to investigate sea cucumber here, and then sponges, and then sea nudibranchs, sea stars, and so on. However, if we work on that, and then sometimes occur question related with the conservation and so on, because most of these resources still not yet cultivated, then will be an issue related with the conservation. So therefore, we have also alternative for them, not only related with the conservation, but in terms of the yield, the result from the marine natural product is also still a challenge, because the yield is really low. So we can just, for example, here to get the anti-cancer from the sponges Discoderma here, for instance, the Discodermolite, to get 20 grams of the compound. So we need to collect, to harvest 1 ton of sponges. Of course, it's not conserved if we talk on the sustainability. Fortunately, that this sponge, for instance, as the most productive marine natural products in terms of the bioactive compounds. So we have also marine microorganisms that living together with this, and some reports mention that actually the microorganism living in these sponges also produce similar compounds that have from the sponge. So this is one of the other. So one of that we isolate fungi, for instance, from the sponges, and also from other resources. For instance, we have mangroves, algae, and then also from the drifting wood, we can also collect this marine fungi. So related with the fungi that we collected from the marine environment, actually it's not different from those that we collected from terrestrial environment. However, the size of this fungi is microscopic. So this is one of the challenges, not like the mushroom that we can easily recognize what kind of mushroom that we have. But for the marine fungi, since it's tiny, so we have to deal with that related with the biodiversity and also with the bioactive compounds from this fungi. So in general, that marine fungi is not different from the terrestrial fungi. However, we have to have the strategy how to get fungi that quite interesting. For instance, using the house that we use to isolate the fungi. So of course, we try to get the fungi from, let's say, interesting area, for instance. So not from the place that probably high polluted, for instance. So this is also one kind of the strategy, because the production of bioactive compounds is also related with the environment where the fungi live, for instance. So we, not only the area and also the house diversity, and also during the cultivation. So we have to also pay attention related to the compounds that we want to have. Okay, so the fungi actually have a lot of potential use, not only for the pharmaceuticals, for instance, but it's also other industrial application. But mostly, yes, also related with my background that during my PhD, I did my research in the Institute of Pharmacy. So therefore, most of my work initially related with the use of active compounds for drug development, for instance. So we have like antibacterial, antifungal activity using the marine fungi and other marine resources. So as mentioned before, that actually these resources is not only can be used for drug development, but it's also other aspects. So therefore, for instance, here we screen for the cellulolytic activity, and the other screening also we do on cheminolytic activity and so on, and of course related with the antibiotic activity. So we have here, for instance, just quick observation related with the antibiotic. We can use dual culture, for instance, or antagonistic test, and then we can quite fast to find which one the fungi that most potential for further research, for instance. Otherwise, we have to work with a lot of isolates that probably also not all of that isolates potential for other, either in pharmaceuticals or probably also in biotechnology use. So we have here, for instance, in terms of the antibacterial activity, we can do on mechanism of action of the compounds. So we have here just to make sure that the antibacterial activity of the marine fungi is also related with the action, how they kill the bacteria, for instance. We can see here some of the pictures related with the changes in cell lysis, for instance. So the cells become broken and so on. So yeah, so this is just to show that the fungi have the antibacterial activity. However, even actually the marine resources, including invertebrates and also marine fungi, they have big potential for drug discovery and development. However, this step is really quite long, and also normally related with the cost. They need a lot of money. So since we have limitation on that, and in some cases that, for instance, when we apply for financial support for the research, so they ask what is the products. So if we develop for pharmaceutical drugs, for instance, and then of course take a long time, at least currently like 10 years. So of course then our research will be really a long way to get the product. So then we change the direction into not the drugs, but still related with the health, but we changed into, for instance, herbal or phytomedicine and functional food development. Because with this target, then we can get the product is quite quick in compared to drug development. So here, one of the products that we have, for instance, we developed some food using spirulina, for instance, the microalgae, and then we have also drink and also crackers. Here we add with the seaweed extracts. So we have here for the drink, we develop herbal drink actually, and then we add with the sargassum extract, the brown seaweed extracts, and then we have here tempe, the Indonesian fermented soybean. So normally this fermented soybean, yeah, just 100% soybean, and then we add with the green seaweed, and then it's quite good and also tasty. And this is other products that we develop again using the microalgae spirulina. So we have snack bars, cookies, and so on and so forth. So actually with this, we can develop more products on that. And then, so not only related with the pharmaceuticals, or probably also foods, nutraceutical, and so on. But from our experience, then we can use these three key points for further research and also product development, for instance. So the first, we developing clean production technology by applying clean technology. And then the second, improve the existing process. For instance, with the marine fungi, we can use the enzymes or the biological process, probably, if possible, to replace the chemical process that mostly have negative impact to the environments, for instance. And then the third, we can also using the enzyme to get the high value derivative compounds. I will give some examples on that. So for the first, it's related with the green technology. As you know that in the seaweed industry, for instance, and also fisheries like shrimp industry, we produce a lot of byproducts from them related with the carrageenan extraction, for instance. So of course we have like the solid waste from the seaweeds. Actually, these resources can be developed into value added. For instance, we can using the fungi to do saccharification, and then we can make this product as the raw materials for bioethanol, for instance, or probably also for other products. And then from the shrimp byproduct, we can make into chitosan and so. And then further, I will give some on the value added products. Okay. So probably most of you already aware on the seaweed industry. I know that Philippines is one of the biggest producer of carrageenan. So to give some example that actually related with the seaweeds, not only to get the carrageenan as the main products, or probably alginate or agar, but we can make the byproducts, either the liquid or probably also the solid byproducts into other products. For instance, you can make into biostimulants, and then for the solid waste, you can make as the raw materials for plastic and so on and so forth. So another example is also possible that we can use this waste or the byproduct into enzyme products. So still a lot of things that we can develop from our resources. So here in our group that developed some bioplastic using the seaweeds, and also here is the food package. This is almost similar to a kind of styrofoam but biodegradable. So we use here the byproducts from the seaweed industry and other agriculture byproducts, for instance, from rice straw, and then from what is that? I forgot the name, but the cane, sugar cane, for instance, so we can make it with this picture. Okay, so another potential use of the fungi, we have here some research that we apply on the agriculture, and then in aquaculture, and also for the development of derivative products using byproducts from fisheries industry, for instance, to develop into biomaterials. Okay, so here is some research that we collaborate with our colleague from Faculty of Agriculture. You know that in Indonesia, one of the vital spice is chili. So we have a problem related with the disease in chili, for instance, because of the fungus, tripping, for instance. So our research resulted that actually the fungi that we collected from the marine environments also have the ability to combat this disease. For instance, here related to the anthracnose, so they have also the fungal antifungal. And then another research that we do with a colleague from the Faculty of Poultry, Animal Science. So since the marine fungi also produce bioactive compounds and also other, for instance, like immunostimulant compounds, for instance. So we try also to add the marine fungal extract into the feed that then we give to the old chicken, for instance, here. And the result is quite good. Not only the immunity of the chickens increase, so they are quite healthy, but it's also the growth is quite good. So you can see here some of the data related to the growth rate of the chicken. And yeah, another example on the aquaculture. So one of the commodity that related with the fisheries industry in Indonesia is shrimp. For instance, with faname, and then also, of course, fish. And one of the problem in this aquaculture related with the fibrosis, the disease that caused by the Vibrio harvae. And we have some research using the marine fungi to combat this Vibrio harvae, especially, and other also fish pathogenic bacteria. So we can see here just to screen how the activity of the marine fungal extract. So these three samples, and then here the floral chemical, when we use the antibiotic in the fisheries industry, for instance, and the antibacterial activity is quite good. So therefore, then we apply the marine fungal extract, so we add into the feed, and we give the shrimp and fish after chilling with the Vibrio harvae. and then the this shrimp and fish is also quite good. So they are quite healthy. So it's not affected by the fetal heart during the challenge, and then also the immunity is increased. So our research exposed that some of the compounds related with the activity, for instance, because the marine fungi contain beta glucan, for instance, sterols and okay, so one focus on the seaweed industry, how marine fungi can also contribute on that. So you know that actually the process for carrageenan extraction mostly is a chemical process. However, we can also do the biological process. Even probably the result is not the same in terms of the chemical characteristic of the carrageenan that we have, for instance, but in other aspect, probably the process is more friendly to the environment. So we have here, for instance, the process that we do using the fungi to extract the carrageenan. So here is the results. And then we try to make this carrageenan into capsule shell, for instance. So in this example, here is the capsule that we developed from gelatin, and this too is from carrageenan. So this is the one that we extracted using the fungi, and this is the one the chemical process. Anyway, this is one of the opportunity. So of course, still need to be developed. So as the conclusion, not conclusion, so probably the thing that we can do in the future. So we have a lot of resources, similar with Indonesia, Philippines also related with the underwater creatures. We have diversity with a really huge number on that. And then we can also, of course, not only the people from the science or biotechnology, but it's also from other fields, for instance. So we need interdisciplinary collaboration. And of course, for us that doing research, so probably one or two or three experience is not successful. So we have to continue our research. So the next probably we can get some good results. And of course, probably for us that have possibility to support probably from the government or in the context of the university, for instance. So as we can also, as the either president or dean, for instance, also have to support staff like us as the researcher to do good research. So yeah, I just like to take this quote that individually we are one drop, but together we are an ocean. So with that, I would like to thank you for your attention, and I give that to the moderator. So please, if any question. Thank you very much.
All right, so thank you so much, Dr. Tarman, for that insightful presentation. Now we'll move on to the Q&A session. This is your opportunity to ask any questions you have about the topic discussed. To ensure we address as many questions as possible, please keep your questions concise. You can type them into the chat box or use the raise hand feature if you'd like to ask your question live. All right, so we have here question from Sir Mark Anthony. Thank you, Sir Mark, for your question. Good morning, ma'am. Can we access the record? Okay.
So probably no question. So maybe
I
in the
think,
future, job that also can facilitate the collaboration not only among the alumni in the Philippines, but probably also the JSPS alumni from other countries, from Indonesia, or probably also not only the JSPS alumni. So maybe with German alumni also we can collaborate.
okay, ma'am.
Yes, actually, sa JSPS with its members is ready for some collaboration, especially those who are in the same field. So we are on the same field in fisheries, so we are looking for future collaborations.
Thank you.
Thank you, ma'am.
Hello, Dr. Tarman.
Yes?
And this is Mark Joseph Rafael from Aurora State College of Technology.
Yes, please.
Okay. So I'm interested in research more on the byproducts processing and the active compounds, especially here in the eastern part and also from the Benham rice, the Philippines. So we are interested in more collaborations in the future. If our president, Dr. Renato Reyes, tells us to. So thank you very much, ma'am, for the very good presentation.
Yeah, thank you. Thank you very much.
All right, so we have here a question from Sir Lito Apello.
Yes.
Ma'am, allow me to read. Considering the distinct ecological roles of fungi, how might their enzymatic activities differ, and are there any specific adaptations that might be observed in each group to facilitate their survival and function within their respective habitats?
Yeah. Okay. Thank you for the question. So we have, for instance, some fungi that we isolate from drifting wood, or mostly we call as liquiculous fungi. So one of the example of the activity that we do in our research is for the cellulolithic activity. So probably also this is related with the ecological role of the fungi in the environment. So we can also, so this one example, yeah. So because you know, in drifting wood, of course they need cellulase for that. So it's related with the cellulolithic activity is quite similar. And other things, one of the examples that we isolate the red pigment from the fungi, that we isolate the fungi from red seaweed. So this is also one of the examples. So in my research before that Silaria, the one that we isolated from the red fungus, Capapicus alvarezii. Also in some media, they also possible to produce the red pigment. So this is one of the examples. So that's why the host of the fungi also will affect what the compounds or probably also the enzyme that produced by the fungi. And related with the survival, so marine fungi divided into two groups actually. One is obligate, the one that only can grow in the high salinity of the media, and then the second is facultative fungi, the one that can be living in freshwater or marine media, for instance. So we can also use this kind of strategy related with the cultural condition to screen in which condition that this fungi can produce stronger or better biological activity. I hope that answered your question. The just of the example that we have done. So probably also possible that we can do other observation related how we can relate the environment and also the cultural condition, for instance, related to bioactive or probably also the enzyme that produce the fungi. Thank you.
Okay, ma'am, thank you. Sir Lito, is that okay with you? So if you have follow-up questions, sir, kindly type in the chat box. Or just raise your hand po.
Dr. Tarman, we have another question here in the chat box. Please allow me to read it for you. Good morning, Dr. Tarman. The marine fungi are harmful as land fungi?
Okay. Yeah, thank you for the question. So actually, yeah, as I mentioned that possible we isolate terrestrial fungi from the marine environment. So if related with whether this fungi harmful or not, so we have no research on that, but some of the strategy how that we can get the fungi known as safe. Normally, we can also isolate the fungi as the endophytic fungi, the one that living in the plants, in the leaf, for instance, that these plants or leaf no indication that we are have any symptom of disease. So if we collect this or use the endophytic fungi, normally this fungi is also not harmful or not one of the pathogenic fungi to the land environment. Yeah, another question related to fungi can be used in other types of plant in the capsicum family. So far we have one research on that. So we don't know yet whether also possible to use to other chili types. Yeah, probably also yeah, can be maybe it's also possible to be used on other species.
All right, so due to our limited time, we will only accommodate a few questions, but then, okay, so however, rest assured that any questions unanswered will be facilitated and sent to our speakers via email. So I think that would be our last questions for our first topic. Thank you so much, Dr. Tarman.
Thank you very much. So since I have another agenda, so I will leave first. So probably later I will join the webinar. It's really interesting. So I would like to join. So again, thank you very much.
Yes, ma'am. Please allow us first to show our appreciation, ma'am. So to show our appreciation to our speaker for sharing her invaluable knowledge, we will now proceed with the awarding a certificate to Dr. Tarman. So allow me to read the text of the certificate. Republic of the Philippines, JSPS Alumni Association of the Philippines, in collaboration with Central Luzon State University, Science City of Muñoz, Nueva Ecija, Certificate of Appreciation is awarded to Dr. Coscheria Tarman for her valuable contribution in providing her technical expertise during the JAAP webinar series 2025 with the theme exploring the diversity and wonders of marine environment, organized by the JSPS Alumni Association of the Philippines, or JAAP, in collaboration with Central Luzon State University, CLSU, on March 5, 2025, given this 5th day of March 2025, via Zoom at Central Luzon State University, Science City of Muñoz, Nueva Ecija, Philippines. So signed by Dr. Evaristo A. Obelia, President of Central Luzon State University, Dr. Renato Reyes, President of Aurora State College of Technology, and Dr. Karl Marx Quiazon, the President of JSPS Alumni Association of the Philippines, and also Dr. Yoshio Otani, Director of JSPS Bangkok, Thailand office. Thank you so much, ma'am.
Thank you very much.
All right, that concludes our first lecture for today. Let us move on to our second lecture. Our next speaker is Dr. Y. Mun Loom, Assistant Professor at Mi University. She will be sharing her insights on the harmful of Rhapidophyte Chatonellia in Southeast Asia, from taxonomy to phylogeography. Dr. Loom, the virtual floor is yours.
Thank you for your introduction. Let me check whether you can see my slides. Do you see? Okay.
Yes, ma'am. It is visible part.
Okay. Thank you for your introduction and giving me this opportunity to share a little bit of my experience and research from my journey in Malaysia to Japan. So today I will mainly introduce my PhD research on the harmful Rhapidophyte Chatonella in Southeast Asia, from taxonomy to phylogeography. Okay. So a little bit of self-introduction. I'm actually a Malaysian from the capital of Malaysia, Kuala Lumpur, and I have gotten my undergraduate. I have gotten my bachelor's degrees in the Borneo site in Kota Kinabalu in Sabah, in the University Malaysia Sabah, with bachelor in marine science from 2012 to 2015. After that, I returned to the Peninsula Malaysia to get a PADI dive master training for 1 year while applying for the Japan Max Scholarship to further my study in Japan. So in 2017, after I get the scholarship from Max, I've continued as a research student in the University of Tokyo for a year to prepare for the entrance exam and so on, to learn Japanese and so on. And finally, I've got my master in 2020 and my PhD in 2023. After that, luckily I got a JSPS postdoc, sponsored by JSPS postdoc for a year and a half. And last year, September, I've landed in this permanent position in MIA University as an assistant professor. So during my undergraduate study in UMS, University Malaysia Sabah, my research topic is actually biotechnology related, which is the development of nanosensor using enzymes for the formaldehyde detection in fish. Because apparently there are some concern about the use of formalin on fish in Sabah. So for my postgraduate journey in Japan, I've continued master and PhD study in the same university of University of Tokyo, in the Graduate School of Agricultural and Life Sciences. The whole study was sponsored by Japan Max Scholarship through the embassy recommendation. And the research topic for my master is the ultrastructure and phylogeny of a new dinoflagellate, Dactylodinium arachnoides, from Sagami Bay, Japan. And for my PhD, I've continued the study of using the same technology, same techniques, continue the study to taxonomy, phylogeography, and ecophysiology of Chatonella in Southeast Asia. Of course, there's not only research, but I also had fun of encountering the real snow for the first time in 2017. And also thanks to my supervisor, Dr. Iwataki, also Dr. Gary Benico's supervisor. Here's Dr. Iwataki. And also there are some researchers from Malaysia and other researchers from Japan. And for my postdoctoral period in Japan National Fisheries Research and Education Agency, it's only a short period of a year and a half. And this is sponsored by the JSPS Postdoctoral Fellowship Standard for foreigners. And the research topic is about the diversity and distribution of Cariniaceae around Hokkaido. This is a picture of the main research facility of FRA in Yokohama, Japan. It is quite new and a lot of good experiment equipments. And why about the research topic, why Cariniaceae has been focused on Hokkaido is because in 2021, an unexpected red tide of harmful Carinia saliformis occurred in Japan. And this Carinia saliformis species has never been reported in Japan before. And unexpectedly in 2021, they formed a very big bloom and caused tons of sea urchin kills and fish kills. So this is a satellite image that estimated the abundance of Carinia species during that bloom. I will further introduce these research topics later. And last September, I've joined Mie University. Probably you don't know where is it. So Tokyo is around here and Osaka is around here. So Mie is in between these two, but is much closer to Osaka or Kyoto. And in our university, we have a big cruise training vessel for students to practice what kind of research we can do on the cruise, and also do some sampling of shores with by using the CTD and equipment that can collect seawater samples from different layers of seawater, and also all kinds of sensors for water temperature analysis or chlorophyll A or salinity, that sort of parameters that we can measure. And in our laboratory, we also have a small boat for monthly survey, which is managed by our laboratory. So each month we went to the coast of Mie to sampling for our research. So the main topics today is my PhD research that looks on the harmful rhapidophyte Chatonella. So Chatonella is a marine phytoplankton that they belong to the Heteroconta, or what we call Straminopiles, where they have two heterogeneous flagellates emerge from the apex. So this is the light microscopy pictures of the Chatonella. And this is a drawing from what we can see in the light microscope photos. So there's the anteriorly directed swimming flagellum that is actively moving and create the sigmoidal movement. And there is also another flagellum that is posteriorly directed that is trailing from the cell. So for Chatonella, there are some taxonomic problem, but mainly reported cell size were about 20 to 100 micrometer in length. And the nucleus is centrally located, while the chloroplast is peripherally located and golden yellowish color. This is a main harmful alga bloom species in Japan. So the taxonomy have to be clarified to understand their harmful effect and different harmful effect, and also the growth, their growth. Yes. So for the taxonomy of Chatonella, it has a long history. It has been described, this genus has been described since 1936, and since then there have been 7 species described, including Chatonella globosa, Chatonella verruculosa, marina, antiqua ovata, minima, and subsalsa. But in 2029, since molecular techniques is available, these 2 species, Chatonella globosa and verruculosa, has been found to belong to different class. which is the class of Dictyocophyceae. Since then, these two species have been moved to the different class, and the remaining 5 species belong to the Chatonella rhapidophyceae. But further analysis show that the 4 species Chatonella marina, Antiqua ovata, and minima belongs to the same phylogenetic clade, and now they are recognized as a Chatonella marina complex. The varieties in the Chatonella marina complex and Subsalsa are phylogenetically distant from this complex. But the remaining problem is that there is no diagnostic morphological features to differentiate between Chatonella marina and Subsalsa. I will further discuss this later because it's quite complicated. But please remember that there are currently two species in the genus Chatonella. And let's look at the harmful impacts of Chatonella in Japan. This is a graph summarized from the data obtained from Japan Fisheries Agency. So the bar shows that the fisheries damage recorded in Seto Inland Sea and Kyushu waters, while the white bar show that those that are caused by Chatonella. So it has recorded the highest fisheries damage in Japan since 1972 and have been occurring year by year. And it has killed all kinds of fish like yellowtail, red sea bream, salmon, and etc. And its distribution has been recently expanded to Hokkaido since 2015. And looking at the global fisheries damage, it has caused fish kills in Mediterranean Sea, India, Southeast Asia, Japan, East Asia, and also in the US. However, the species identification using molecular data is not as widely analyzed. So in Southeast Asia, East Asia and Southeast Asia, we have Chatonella marina. And Southeast Asia, we have only 2 reports, which is in the Singapore, we have Subsalsa. And in the Indonesia, in the mangrove area I forgot the place, Indonesia, we have Chatonella marina complex. So the problem with Chatonella information in Southeast Asia is that the fisheries impacts and species distribution are still limited. So during my PhD study, we conducted intensive survey along this area and did some molecular identification and also clarify the taxonomy problem. So 3 main research problems addressed in my PhD thesis is that first is to summarize the fisheries damage and species distribution of Chatonella in Southeast Asia and East Asia. Then I tried to find the diagnostic morphological characters that can contribute to the taxonomy by using light microscopy, fluorescent microscopy, and also transmission electron microscopy. And finally, I conducted a ecophysiological experiment to determine the effects of temperature and salinity. So the sampling location is indicated by the yellow dots. Because this research is conducted since 2012 to 2023, but because of Corona, so the coverage is still not enough. So for example, in Vietnam, we still don't have any samples. But with collaboration from Southeast Asia's researchers, we have collected 45 strains, successfully collected 45 strains and 23 culture strains from East Asia, in China and Russia as well. And because of their morphological similarities, so we first conducted molecular phylogeny using different primers to determine different regions of the DNA. And then with the species known, I tried to find the morphological differences between these genotypes and finally conducted ecophysiological experiments. So as a summary of the harmful impacts of Chatonella in Southeast Asia, before my research, there's actually some reports of shrimp kill in Southeast Asia and fish kills in Japan and Russia. But during my research period, I've obtained some very old literatures and also reports in different languages from collaborated researchers in this region. So I've successfully found out that there are actually some more fish kills and shrimp kills in the Southeast Asia. For example, in the Philippines, in 2004, there's a fish kill in Bolinao. And also in Vietnam, there are some shrimp kill in Nha Trang. And also in Thailand, there is also some shrimp kill. While in Malaysia, we have fish kill and a shrimp kills. But there's no, they are reported, in these reports, they are reported as various species of Chatonella, like Antiqua marina or Wata subsalsa or Chatonella sp. But there's no molecular verification to find out the species. Well, if you are interested in the detailed report, you can find my paper in Harmful Algae 2021. And for the molecular identities of Chatonella in Southeast Asia, okay, there are in the LSU rDNA, we can see that there are 5 different phylogenetic clades in the genus Chatonella. So there's Subsalsa 1 and 2, and Chatonella marina in the clade 3. And there's additional 2 new clades that we have no idea what were they in the clade 4 and 5. So the sequences analyzed in this study is indicated by the black dots. And okay. Okay, so both Chatonella subsalsa marina, as you can see, are common in the Southeast Asia. And clade 4 and 5 are found in Malaysia and Thailand. And too bad we don't have enough sampling. We didn't cover enough sampling in Indonesia, Vietnam, and Philippines. So we are not sure that whether they also distributed in these countries. So the next question is that, is these 2 new genotypes, the clade 4 and 5, represent new species? To define a new species, we need to find diagnostic morphological characters in additional to the molecular data, because morphology is, because in the same clade, even though in the same clade, even in the different clade, you don't know where the species definition is being defined. So to define the species, we need to have both of this data. So by looking at the morphology of Chatonella, previously in, I think 1982, there was said that since 1982, these 2 characters are said to be able to differentiate between Chatonella marina and Subsalsa. But let's see. So for Chatonella cell morphology, there's actually 2 layers of the cells. 2 layers. There's the exoplasm and there's the endoplasm. So on the cell surface, we can see that there's some mucus structures with some button-like structures. But in the intracellular structure, we can see the nucleus and also the chloroplast, and also sometimes the mucocyst. So let's look at the oboe-shaped mucocyst. In Subsalsa, mucocyst has been reported since 2001. And in Marina complex, actually, they could not find, but in most of the Chatonella marina complex, but in some of the reports, mucosis has also been observed in the Chatonella marina complex from Japan. So this character is actually not reliable to separate these 2 species. And the next characters is to observe the pyrenoid region attached to the chloroplast to see whether the thylakoid penetration is present in the pyrenoid. So in Subsalsa, it is obvious that the thylakoid penetration has been observed in the pyrenoid, while in Chatonella marina complex, this structure is ambiguous. And okay, let's look at the light microscopy of these 4 genotypes first before we go into the transmission electron microscopy. So for Chatonella subsalsa, marina, clade 4 and 5, they have similar or overlapping shapes, such as the lemon shape or elongated or ovoid shape. And to look at the cell size range, the blue shows the Chatonella marina cell width and length. So Chatonella marina complex has, and these strain names are shown by this, for example, GBIN7. So Chatonella marina complex actually have a very big size range from 25 to almost 90, 80, 82.5. And then Chatonella subsalsa in rates shows a smaller range, but still overlap with some of the Chatonella marina. And the clade 4, which is shown by the pink data, showing overlap cell sizes with Chatonella subsalsa, and also clade 5 has a smaller size, but also overlapping. So this cell size range actually is not informative to separate between these four genotypes. And to look at the chloroplast autofluorescence, we can determine the cell shape and cell number of these four genotypes. As you can see, Chatonella subsalsa has discoidal shape and about 140 chloroplasts counted. And Chatonella marina has also similar shape with about 192 chloroplasts. While in clade 4 and 5, the chloroplasts are somewhat different, where the clade 4 has elongated and slim chloroplasts with a much lower number, while clade 5 has a bean-shaped curving bean-shaped chloroplast, which has a similar number. So the chloroplast shape and number was slightly different, but still not enough to differentiate between these four genotypes. And for the oboe-shaped mucosis, we used a chemical called neutrarate to stimulate the release of this mucosis to confirm whether they have they contain this mucosis and can release them from the cell. So for subsalsa, we can see that there are a lot, and marina also. Clade 4, this is mucosis from the compressed cell without using the neutrarate stain, but the presence of mucosis has been confirmed. And in clade 5, we also can see some little mucosis released from the cells. So all four genotypes actually have the oboe-shaped mucosis, and it is not enough to separate them. And next, we will look at the thylakoid penetration into the paranoid using transmission electron microscope. So this is the chloroplast, and a part of it is the paranoid. So in subsalsa, we confirm that there is single-stranded thylakoid penetration into the paranoid. While marina, we have also thylakoid penetration. Also in clade 4 and 5, we also can see thylakoid penetrations into the paranoid. So same, all four genotypes have the thylakoid penetration into paranoid. However, the patterns of the thylakoid penetration appears different. So in subsalsa, we have single-stranded. Marina, we have 1 to 3 branching thylakoids. And clade 4, we have 1 to 3, similarly 1 to 3 branching. And also we found single tubular thylakoids in the paranoid. So for clade 5, we have 1 to 3 branching thylakoids in addition to a heavy lamella, which is the oppressed thylakoids in the paranoid. So the number and patterns of thylakoid penetration could be said to be different between these four genotypes. So now we have found the characters that can differentiate between these four genotypes. So in summary, for the diagnostic morphology, the ultrastructure of paranoid is useful to differentiate between these four species, which is the number of thylakoids, the shape of the thylakoids, and the patterns of penetration. While transmission electron microscopic techniques are time-consuming and difficult if you don't have the equipments and the techniques. So what I suggest in my PhD dissertation is to differentiate them using number and shape of chloroplasts, and also supported by molecular data whenever it's possible. So based on these characters, the two clade 4 and 5 were described as two new species, which is the Chatonella tenuiplastida, named after the slim chloroplast, tenuis plastida, and also Chatonella malayana, which is found in the Peninsula Malaysia, which is previously called Malaya. So to look at the phylogeography of Chatonella in Southeast Asia, of this sampling location, we found that there are 4 species in the Southeast Asia, while there's only Chatonella marina in the East Asia. And to look at this distribution of Chatonella species in Southeast Asia, we can see that there are some overlapping. For example, in Thailand, we have Chatonella tunicplacida and also Sapsausa, while in the Peninsula Malaysia, we have Malayana and also Marina, and here as well. So I wonder whether they have different ecological niches because apparently they have similar distribution. So they must have some different preference in the temperature or salinity or other abiotic factors. So I conducted temperature and salinity using different treatments. So for temperature, we have temperatures with a 2.5 degrees Celsius differences. And for salinity, we have 5 treatments with five salinity 5 differences with a fixed salinity or temperature. So this experiment was conducted until day 20 to calculate the specific growth rate in different temperatures and salinity. And all these culture strains were used from those collected from Southeast Asia. So let's look at the effects of salinity. So the red show the results from Chatonella subsalsa. The blue is Marina, pink is Teneplacida, and green shows the Malayana. In the low salinity, only subsalsa shows steady growth, while Marina shows like okay growth, and Teneplacida and Malayana show no growth at all. But in the highest salinity, all these 4 species has grown well, but Subsalsa grew better than all other species in all salinities. For the effects of temperature, we have 10 temperatures. So let's look at the lower temperature. So in the lower temperature, 23 degrees Celsius or less, Chatonella marina shows a better growth compared to other species. Whereas in the higher temperature, Chatonella subsalsa shows unexpectedly good growth compared to other species. More surprisingly, in 35.5 degrees Celsius, which is very high, only subsalsa show growth. While the 2 new species show a very limited range of temperature where they can grow. So in conclusion, Marina has higher growth in lower temperature, while Subsalsa has higher growth in 25.5 degrees Celsius. And Teneplacida and Malayana shows growth only in higher temperature, but not too high. And to summarize the temperature ranges, I've drawn this summarized graph. So the box shows, the arrow shows the growth temperature range, but whether they can bloom in those temperature, which what I define as grow more than 10,000 cells per milliliter, were indicated by the shades. So Subsalsa shows a bloom temperature range of 25.5 to 33, while Marina has shown bloom temperature in the lower temperatures. So their growth are slightly overlapping, but the bloom temperatures are somewhat different, indicating they have different ecological niches and might bloom in different environmental conditions. And to summarize phylogeography and their bloom harmful impacts, I've overlapping the fish kills map and the species distribution map. So in the shrimp kill area and also 2004 bloom fish kills, Subsalsa shows the overlapping distribution, and also because of their high growth, it is possible that Subsalsa is the potential causative species in these areas. And in Peninsula Malaysia in 2016, Chatonella malayana was actually isolated during a bloom with wild fish kill in 2016, which suggests etotoxicity, even though the growth from my experience was not so high. It is still possible to form fish kills. And while for Chatonella marina complex, there's only Chatonella marina complex in the East Asia. So I have tried to analyze why this could happen. I compared to the annual average of sea surface temperature using satellite data from 2012 to 2019. And this line here shows that 23 degrees Celsius might be the border line for the expansion of Chatonella marina complex. So because they can survive and grow in low temperatures, it is possible that is why they can be spread to higher latitudes, to the colder waters. But looking at the global pictures, 23 degrees Celsius collect the borderline are plotted like this. And marina is really could be found in these regions. Whereas for Tenipacina marina has been newly described, so we don't know its distribution along over the world. But for Chatonella subsalsa, we can find also the subsalsa in the Mediterranean Sea with lower temperatures. However, these subsalsas belong to clade 1, and while these subsalsa belong to clade 2, so the relationship between the subsalsa clade 1 and clade 2 still remains unquestioned, unresolved, because the lack of samples and the lack of studies in these regions. So hopefully someone will do the research or provide the samples for further analysis. And there's also some more distribution of Chatonella that they have known without the molecular data. And these places have been reported with fish kills, so we still don't know what species caused the fish kill. And to continue to my current research, it is not Rhaphidophite anymore. It is mainly on the dinoflagellates, where it is one of the most diverse group in the marine phytoplankton. And their general shape morphology looks like this, where they have transverse flagellum along the cingulum, and also they have longitudinal flagellum directed posteriorly. So these are all pictures of the dinoflagellates, and they range from 2 micrometer, as small as 2 micrometer to 2000 micrometer in length. And they contain photosynthetic, mesotrophic, or heterotrophic species. And this group also contains many toxic and harmful species, which is of many of deep interest. So during my JSPS postdoc, I have conducted harmful algal bloom study in Hokkaido, mainly on Cariniaceae. So in Hokkaido, there are mainly paralytic shellfish poisoning and diuretic shellfish poisoning causing toxic dinoflagellates, such as Dinophysis alexandrium. They have been recorded since the 60s. They have been regularly monitored even now. But since 2015, the harmful algal blooms of the Carinia mikimotoi has been detected in Hokkaido. And in 2021, another species, Carinia saliformis, has also occurred and caused severe fisheries damage in Hokkaido. However, the Cariniaceae distribution in Hokkaido is understudied because no one expects them to grow in such cold temperatures. So what I've done in my JSPS postdoc is using a 10-year samples collected in Mombetsu, this red square here, that to analyze the distribution or the occurrence pattern of Cariniaceae in this region. So Mombetsu is actually an interesting place because it is affected by 2 main currents, which is the East Sahalin Cold Current from November to March, where the nutrients are abundant. And also in the summer, it is affected by the Soya Warm Current, which is not so nutrient rich, and but it is warm. So using 10 years weekly sampling data, we have analyzed the metabarcoding using environmental DNA and also the abiotic parameters such as water temperature, salinity, chlorophyll A, nitrate, and phosphate and nitrate to try to determine the species diversity and distribution of Cariniaceae around Hokkaido. So for the results, we have from the 448 samples using 3 different primer sets, we have found out that there is at least 19 Cariniaceae in Mombetsu, but most of them are Calodinium species, more than about 90%, while Carina, Mikimoto, and Saliformis are only very rarely detected. Uh, However, using this 10-year data, there is no obvious correlation between the occurrence patterns and also the environmental parameters. So probably Monbetsu data is useful for long-term analysis, although maybe further detailed analysis is needed. And last year, since I have joined MIE, so I have focused on this region because it is a very rich area for the production of marine products like scallops, abalone, and oysters, and also lobsters and other fishes. But this area actually have very frequently occurred harmful algal blooms. So for example, in this region, in Gokasho Bay and Ago Bay and Madoya Bay, there is actually the first description of many harmful algal species. Their type locality belongs to this interesting area. So for example, Carinia mikimotoi, as I mentioned, and also Akashiwa sanguinea, which is also very common in the Southeast Asia, and Heterocapsa circularis squama that can cause shellfish kill. So from now on, my research will be focusing on determining whether there are other harmful species in this region, and also what is their occurrence pattern and the related environmental parameters. And also because this kind of they call this type coastal area, it is very easy to accumulate the harmful algal bloom species cyst, which is the part of the life cycles. So this cyst abundance has also been is also needed to be clarified, and also the effects of fisheries of this species in this area. So with that, I would end my presentation with some take-home messages. So I have started my postgraduate study with taxonomy, and it doesn't end there because taxonomy is the important field that can contribute to the other studies like ecophysiology, phylogeography, harmful impacts, and occurrence patterns. But it is most how to say the basic research that must be clarified before we can continue to the other studies. And for students that are planning to go continue to further research, please do not be disappointed when the scholarship you apply that didn't get through. Try again because persistence is the key to success. And if you're interested, you can visit our homepage of our laboratory. Unfortunately, it's only in Japanese, but you can see all the pictures that what kind of activities we are conducting in our laboratory. Thank you for your attention.
All right. Thank you, Dr. Loom, for sharing your valuable insights on harmful algal blooms. Your presentation has shed light on the understanding the health risk to recognize the signs of harmful algal bloom. Your expertise has provided us with actionable knowledge to protect ourselves, our communities, and our ecosystem. Thank you so much. We are now ready to address question. And with that, we are now read questions for you, Dr. Loom.
Okay.
For our first question, what is the effect of these organisms in the fish physiology resulting to their death? Is it due to some poison from these organisms or as a result of low DO level? DO level as a result of their massive production bloom in a certain area. How about its effect on muscle?
Thank you for your questions. So actually, it is possible also that the harmful algal bloom can cause low DO that can cause the fish kills. And also there is also possibility that this harmful algae can form toxins or other metabolites that can affect the fish or shellfish. So depending on the species and also depending on the condition that the species grow, we have to analyze the harmful algal bloom waters, and also the cause of the death of the fish by doing more detailed analysis. So I am not sure how to answer your questions because It depends on the species and the conditions.
Thank you, ma'am. So we have here another question. So hi, from Miss Christine. Hi, ma'am. Did you use the new bore for identification and counting the following microalgae?
Sorry, what do you need? New bore?
New bore, ma'am.
New. I'm sorry, I didn't catch the word because I could not see the chart.
Yes, ma'am. Maybe we can ask Miss Christine. Could you please
Hello, ma'am. Excuse me. Can you hear me?
Hi. Yes.
Oh, hello. My question is that did you use the new Bower to count the following microalgae as well as the identification of the microalgae?
Neubauer, no, I did not use the Neubauer. And for counting, we use the most traditional way, which is the Sedgwick rafter, and observe under light microscopy for counting.
Okay, thank you so much, ma'am.
Yes.
Thank you so much. Another question from our participants. In terms of HAB dynamics, how might the presence of these diverse clades influence the frequency, intensity, and duration of Chatonella blooms in Southeast Asia? And what are the potential consequences for marine ecosystem and aquaculture?
Okay, thank you for your questions. So for Chatonella, there's also known that they will form cysts that can last in the sediment for a very long time. And when the conditions allows, this cyst of Chatonella actually will germinate and contribute to the Chatonella cells in the seawater. So to know the Chatonella distribution in each area, we can know that whether they are there and first the presence, whether they are there, and then we have to determine what kind of environmental factors that can promote to their growth, and also what kind of conditions that they will release some harmful metabolites or chemicals that can kill the fish. So to answer your questions about the hub dynamics, so this presence is the first step to know whether Chatonella will affect the fisheries in Southeast Asia. Then further study of the ecophysiology, for example, or the field observation, long-term field survey to determine whether their presence are always there or is it seasonal. That kind of questions that need to be answered. Yeah. So the potential consequences for marine ecosystem and aquacultures. Yes, if they are there, there's the potential that this Chatonella will cause us our marine fishes when they form blooms. Yeah, I think that answers the questions.
All right, thank you so much, Doctor. I'm sorry and apology to our participants because we have only limited time to accommodate this question. However, rest assured that any unanswered questions will be facilitated and sent to our speakers via email for a shared documentation.
Okay.
To show our deepest gratitude for you being one of our speakers, please allow us to award this certificate.
Thank you.
Please allow me to read the citation. Republic of the Philippines, JSPS Alumni Association of the Philippines, in collaboration with Central Luzon State University, Science City of Muñoz, Nueva Ecija, Philippines. Certificate of Appreciation is awarded to Dr. Y. Munlu for her valuable contribution in providing her technical expertise during the JAAP Webinar Series 2025, Exploring the Diversity and Wonders of Marine Environment, organized by the JSPS Alumni Association of the Philippines in collaboration with Central Luzon State University, CLSU on March 5, 2025. given this 5th day of March 2025 at Central Luzon State University, Science City of Muñoz, Nueva Ecija, Philippines. Signed, Evaristo A. Abella, the President of the Central Luzon State University. Signed, Renato G. Reyes, President of Aurora State College of Technology. Signed, President, JSPS Alumni Association of the Philippines, Dr. Karl Marx Kiazan. And signed, Yoshio Otani, Director of JSPS Bangkok Office in Thailand.
Thank you.
Thank you so much.
Thank you for this opportunity. I'm sorry I have other agenda, so I have also leave this seminar. So thank you very much for having me. It is a great chance to see all of you. So hopefully we can meet in person next time. Thank you.
Thank you, ma'am.
Thank you.
Okay, so we are now moving forward with our third lecture. We have with us Mr. Marco Flores de Guzman and an assistant professor at Aurora State College of Technology. He will discuss seagrasses along the coast of Dilasag, Aurora, Philippines. Mr. de Guzman, you may now start po.
Okay, may I share your next slide presentation? Okay, so good morning to all of you. So let me first thank you, the GSPS Alumni Association of the Philippines, for organizing this webinar. So my topic is all about the seagrasses along the coast of the Lisag, Aurora, Philippines. So for this session, it will be divided into three parts. So the first topic, it will be what is seagrass and why is important. The second will be focused on the seagrasses along the coast of the Lisag. And lastly, I will share some insightful ways on how we respond to our findings and future plans. So my first question is, do you like to eat crabs, shrimps, and sushi in restaurants? Or do you like getting on the beach and smell the salt on the gentle sea breeze? And do you want to see some marine organisms like marine turtles and dugongs? If you say yes to one of this question, then today I will introduce to you the heart of the ocean, the seagrass. So, what is seagrass? It is an aquatic or angiosperm, meaning they are a flowering plant that lives totally in the water, and they are a true plant, and they photosynthesize like the plants in the terrestrial land. Originally, they do not totally live underwater, and seagrasses evolves to live from the land and completely live submerged in their marine environment way back to the Cretaceous period. So in terms of its importance, it serves as nursery and hatchery for our commercial fisheries. And almost all of the seafoods we eat is tied directly by spending a portion of its entire life in the meadows or indirectly by eating something that coming from the seagrass meadows. Recent estimates suggest seagrass meadows support the productivity of 20% of the world's biggest fisheries through nursery habitat provision, and studies also shows that about 40,000 fishes and 50,000 invertebrates can be supported by the 10,000 tons of leaves produced in an acre of seagrass beds. Seagrass is also one of our secret weapons to combat climate change. Seagrass done it by taking carbon to their bodies from the environment and release oxygen. In addition, seagrass also store carbon by sequestering the carbon and placing it to the sediments where it can remineralize back to the carbon cycle. So here, as you can see, is a graphical representation on the carbon storage of the different ecosystem and their corresponding carbon deposits. As you can notice, coastal blue carbon ecosystem can store 2 to 5 times more carbon in their soils per unit area than the terrestrial ecosystem. In seagrass alone, it stores almost 600 megaton of carbon per hectare and almost 2 times than the tropical forests. These characteristics makes this ecosystem extremely valuable as nature-based solutions for mitigating and adapting to climate change. However, the great challenge for seagrass ecosystem is that they've threatened globally, with evidence indicating accelerating rates of loss and degradation. Their common shallow water presence at the coastal land interface makes them highly vulnerable to disturbance and anthropogenic impacts. As we all know, the fact that the interest of most agencies mainly on coastal resources with immediate economic value or impact like seaweeds, coastal development, and others. And that's the reason why seagrasses ecosystem is neglected. So here at ASCOT, we are working to evaluate the species of seagrass and evaluate its state for mitigation and management. So our work is focused in the municipality of Glasag, the first phase of our project in the northern Aurora. So the municipality of Glasag is located in the farthest part of the Aurora province near the Philippine Rise. It is one of the municipalities that characterized with rich marine biodiversity and produce tons of tuna and lobsters. So our team gathered data by using the procedure by Mackenzie, 2001, that uses a 50-meter by 60-meter transect line that will be laid in the seagrass area. The sampling procedure is that we have 3 stations that have 3 transects, and every 5 meters we lay a curve that measure a 50 by 50 centimeter at 10 intervals. And we also use the seagrass watch guide for the photo comparison rather of the seagrass cover. So here is the example of a quadrat laid every 5-meter interval. So we use a 0.5-meter steel quadrat of the transect steel quadrat, and the data gathered will be recorded including the coordinates. And for identification of the potential traits in seagrass, we assess the anthropogenic activities that could evidently affect the seagrass and the macro invertebrates in the area. We also interview local and what are the things that they observed for the past years in the area. We also conduct ocular inspection and geotagging of these potential traits. So here we have found that the seagrass beds cover a total area of 8.7 hectares. The largest is in the Barangay Masagana with 7.4. This was followed by Barangay Jagyan with almost a hectare. And in the area with the least seagrass cover is the Barangay Dinyog with 0.24 hectare. In terms of the seagrass species, we have found 4 species of seagrass. First is the Inhalus acoroides. It is only the species of the seagrass that does not aerial surface pollination, in which the pollen and the styles remain dry. In Inhalus is surface pollinated with male flowers that detach from the plant to float on the surface until they reach a female flower for where the pollination occur rather. So it's also considered a climax species, meaning that they are the most established species in the area. Second that we have identified is the Thalassia himprichi or the Pacific turtle grass. It is the favorite diet for our marine turtles, and its growth rate increases with carbon dioxide enrichment. So it can tolerate lower light conditions that caused by algal blooms and allowing it to respond positively to ocean acidification and other disturbances. Another species that we identified is the Syringonium istophilium. This or what we call the noodle seagrass. The shoots are encased with a sheet that base with each consists of 2 or 3 hollow that have tubular leaves with small pointed tips. And in calm waters, the shoots may be long and branch. We also identified the Haloduli pinifolia. It grows in a sandy or muddy substrates from upper littoral or sublittoral areas. It's ephemeral with rapid turnover and high seed set, and is well adapted to high levels of disturbances. This species can grow rapidly and is a fast colonizer and often heavily exercised. Unfortunately, way back 2019, we have already identified 5 species of seagrass. Yet, in 2023 rather, we only identified 4 seagrass species. And the species of Haluvilla ovalis have been missing. So this might be the result of anthropogenic activities in the area, or This may be attributed to the fact that there were species of seagrass that germinate and occur in a period of a favorable drought. In terms of its seagrass cover in the area, among sampling stations, Barangay Dinyog has the highest seagrass cover with 47%, and this was followed by the Barangay Masagana with 43. That is the more diverse seagrass species, had more diverse seagrass species, and also the Barangay Jagyan with 37%. In average, in terms of its mean comparison cover, the average seagrass cover in the municipality of Dilisag is 42%, and the condition equivalent based on the criteria of Fortes is fair. And it's lower compared for the areas in the country like Cape Bolinao with 75%, in Puerto Galera with 95%, and in the Honda Bay with 90%. The low seagrass cover in the area may be due to the anthropogenic activities that was observed during the assessment that the fishermen anchored the scrape, the seagrass meadows, and uprooting the seagrasses in the area. Recent reports also showed that these anthropogenic activities affect the seagrass percentage cover. Therefore, it is imperative to consider strategies to mitigate those anthropogenic activities for the seagrass to recover. In terms of its seagrass density, it is also noticeable that the Barangay Dinyog has the highest density with 523, followed by the Barangay Jagnan and Barangay Sagana. We also have identified macroinvertebrates. There were crabs like Portunus sagulentus. We have also the Portunus pelagicus and Clibanurius tricolor. And also in the shells in the area, we have also identified the Turbo, Chrysostomus, Nirita, Peloronta, and Euspera fosca. We also have identified the Anadara racemosa and Artina selendica. In terms of the potential traits, as we all know, seagrass meadows' presence in the intertidal area of coastal land makes them highly susceptible to disturbances and also anthropogenic activities. Recent studies have shown that overfishing, ocean acidification, sedimentation, increased water temperature, land reclamation, boating, and aquaculture are significant threats to seagrass worldwide. So we have documented the different potential threats to the seagrass in the area. So first is that the port of the fishermen. We have observed that the seagrass in the area have been scraped by the boats in the area. As we all know, those boats are equipped with anchor, and when they port, they usually tend to drop their anchor, and this eventually drag the seafloor and eventually uprooting those seagrass that could lead to the desolation of these plants. Another one is plastics. This affects the macroinvertebrates in the area as they might ingest and evaluate their physiological aspect of production. Another one is the chemicals when they are constructed or use chemicals, and this might affect the physiological aspects of seagrass and macroinvertebrates. Another one that we observe is that the household effluent. This effluent is harmful because it causes high organic content in the area, and it might cause phytoplankton bloom that disrupts the photosynthetic activity of the seagrass in the area because it serves as we all know, it serves as the blockage of the sunlight. Another one is the gleaners of the shells in the area. Some of the fisher folks in the area disturbs the seagrass by like walking around them, and some digging up the seagrass, eventually uprooting those seagrass. It also affects the seagrass in the area. And also the chemicals used in farming. We have observed that most of the farmers in the area uses high or chemicals rather and high nutrient fertilizers that during somewhat rainy season, these chemicals will tend to run off in the seagrass area and eventually affects them. So with that, so after evaluating the area, here we have taken actions in order to mitigate those problems that we have identified. First is we conduct environmental awareness in order for the community to become fully aware and mindful on the importance of seagrass meadows. We also want them to have that sense of urgency in caring the seagrass meadows and become the stewards of this environment. This is getting by them involved firsthand in the coastal planning up to the main into the implementation of mandates, which will increase the sense of urgency in managing the coastal environment. We also aim to establish a marine laboratory that will be used to become the center for the restoration of the seagrass beds in the province. By developing and adopting mature technologies or practices rather for the seagrass restoration projects, and also our longer aim is to establish three restoration hubs throughout the province in order to fully maximize the restoration process that we want. This will be located in the three strategic municipality of the province, which is in the northern, in the Kasiguran, in the central, which is in the Baler, and the southern area, which is in the municipality of Dingalan. So the month of March also is considered as a seagrass awareness month. So it is very timely that I have been given the time to share to you to raise awareness about the significance of seagrass in our life. So with that, if you have any questions or for future collaboration, so here is my details, and that ends my presentation on the seagrasses along the coast of Dilasag, Aurora. So thank you very much.
All right, so thank you so much, Sir De Guzman, for pointing out the vital contribution of seagrasses to maintaining a balanced marine ecosystem. So may I just share? Earlier we have learned about Chatonella blooms, and according to Inaba et al. 2019, certain marine bacteria associated with seagrass beds can actually suppress Chatonella blooms, and this bacteria act as a natural control mechanism by limiting the growth of harmful algae. So restoring or introducing seagrass beds could enhance the presence of this bacteria, beneficial bacteria, and providing an eco-friendly solution to mitigate these blooms. So thank you so much once again, Sir Marco. So please allow us to extend our appreciation.
Thank you.
Okay, so yeah, we're going to move on sa ating question and answer session. So allow us to read the questions for you, sir. So how does the restoration of seagrass meadows help increase the resilience of coastal communities to climate change, particularly in regions vulnerable to sea level rise and extreme weather events? And how does this support SDG 11, or the Sustainable Cities and Communities, and SDG 13, Climate Action?
Okay, so seagrass helps on the mitigating of our climate change by sequestering the carbon in the atmosphere. So they trap the carbon in their sediments. So they prevent it by remineralizing back to the carbon cycle. So it helps in terms of carbon sequestration, and also uh, comparing it to the terrestrial lands or trees. They store two times more carbon in the cycle.
Okay, so another question. Considering the role of seagrasses, does it influence the composition and diversity of microbial communities, sir?
For that, we don't have any studies about the diversity of the microbial communities in the seagrass area. But I think this is a good idea also to tackle in order or research gap to understand or to focus. So I think in terms of the seagrasses, it can influence it can really influence the composition of the microbial communities in that area of the seagrass area.
All right, so I think that would be our last question. So please allow us to extend our appreciation to your lecture, sir. Okay, so allow me to read the citation of the certificate. Republic of the Philippines, JSPS Alumni Association of the Philippines, in collaboration with Central Luzon State University, Science City of Muñoz, Nueva Ecija, Philippines. Certificate of Appreciation is awarded to Mr. Marco Flores de Guzman for his valuable contribution in providing his technical expertise during the JAAP webinar series of 2025 with the theme Exploring the Diversity and Wonders of Marine Environment, organized by the JSPS Alumni Association of the Philippines, or JAAP, in collaboration with Central Luzon State University, or CLSU, on March 5, 2025. Given this 5th day of March 2025, via Zoom at Central Luzon State University, Science City of Muñoz, Nueva Ecija, Philippines. Signed, Dr. Evaristo A. Abella, President of Central Luzon State University, Dr. Renato G. Reyes, President of Aurora State College of Technology, Dr. Karl Marx A. Quezon, President of JSPS Alumni Association of the Philippines, and Dr. Yoshio Otani, the Director of JSPS Bangkok, Thailand office.
Thank you very much.
Thank you so much, sir.
All right, we are now heading to our final lecture. We have Dr. Gary A. Benico, Professor 2 at Central Luzon State University, who will talk about naming the bloom, why taxonomy matters in harmful algal blooms research. Let's give him our full attention. Dr. Gary?
Thank you. Let me share my PowerPoint. Okay, can you see my PowerPoint now?
Yes, Doc.
Okay, good day, everyone. First, I'd like to express my gratitude to the JAAP, JSPS Alumni Association of the Philippines, through its president, Dr. Karl Marx Quezon, for inviting me, okay, to share my research, my experience, okay, working on harmful algal blooms. And also, I acknowledge the support of our president, Dr. Evaristo Abella, and our BPAA is also here, Dr. Lavilina Velasco, and also the president of ASCOT, Dr. Reyes. Okay, thank you very much for inviting me here. Today, I will be discussing why taxonomy plays an important role, okay, in harmful algal bloom research. So, earlier, Dr. Wyman Lam, okay, introduced to you our field. So Dr. Wyman Lam is actually my kohai in Japan when I was doing my PhD. So she was still doing her master's at that time. Okay, because these organisms are very important. Okay, so later on you will see why this phenomenon is actually very important. Okay, so first, before we dive into this topic, let me introduce myself. I'm a professor at the Department of Biological Sciences at CLSU, and I have been involved in hub research for many years, okay, for I think more than a decade. And my expertise lies in the taxonomy and phylogeny of dinoflagellates and other hub species, utilizing various tools such as molecular phylogeny, high-end microscopy, and DNA sequences. Okay, so as I've mentioned earlier, we were mentored. Dr. Wyman and I were mentored by Dr. Mitsunori Iwataki in the University of Tokyo. So after my PhD, I returned to the Philippines, okay, to offer my expertise and to serve my country. Okay, and fortunately, I was able to get a teaching position at Central Luzon State University. So I established my laboratory, my small laboratory, okay, at LSU. So right now we are implementing externally funded projects, and also I am supporting our not only our graduate and undergraduate students, but also other students from Northern and Central Luzon. Okay. Okay. So first, let me define what is harmful algal bloom. So basically, they are just proliferation, okay, rapid population explosion of algae that can have toxic effects to the marine life, seafood safety, and human health. Okay, and they are often associated with red tide. Okay, here you can see this is a red tide that happened in Mindanao. So as you can see, it's blood red. But technically, not all red tide are harmful. Okay, but red tide, there's always water discoloration. There are red tide that are actually beneficial, that are good to fisheries, okay? Because they can actually support marine productivity. But there are some species of microalgae that produces toxin. That's where the red tide become very harmful. It's now called toxic algal blooms, okay? And the term red tide is not always red, okay? Here you can see it's red, it's bloody red, but sometimes it can appear brown, okay? This is another red tide that happened in El Nido, Palawan, okay? So you can see there the separation, the blue water and the red tide affected area. Okay, so it can also appear green. So depending on the species. So this is a red tide of Noctiluca scintillan. That's another species. So depending on the species, the color of the red tide can happen. Okay, you can also have orange colored red tide. Okay, and hubs are actually a major environmental issue. Okay, they can contaminate because they can contaminate seafoods. Okay, because some of the species actually produces biotoxins that can be biomagnified, okay, transferred to seafoods, especially mga shellfish. And also they can cause massive fish kill, either through the biotoxins they produce or through low dissolved oxygen. So once this microalgae bloom, sometimes they collapse, okay, eating up the dissolved oxygen, then that's the time you can have hypoxia and anoxia. That can also cause fish kill and can also alter ecosystems. So it can also kill sea turtles, seabirds, marine mammals. Okay, so they are very harmful. Okay, but hubs are not just an environmental issue. Okay, they are also a major public health concern. So red tide, I think you have heard about red tide. So in the Philippines, there are a lot of poisoning cases that has been recorded in several coastal areas in the Philippines. For example, here you can see that there was a red tide poisoning in Masbate. You have another one in Pangasinan. Okay, way back in 2014, there was one dead and 25 hospitalized. Also, there was another one uh, in Samar, Eastern Visayas. Okay, so basically, this is a public health concern because it can kill. Okay, toxin produced by this microalgae can kill people, can kill business. Okay, that's why it's a very important environmental issue and public health concern. And the impact of harmful algal blooms sometimes can be so severe that local government units are forced to declare state of calamity. Okay, so sometimes baka naririnig nyo yung state of calamity usually are being declared when there is a typhoon, earthquake, but red tide, when they are so severe, LGUs can actually declare state of calamity. Okay, so in this way, when the LGUs declare state of calamities, they can actually mobilize emergency funds, implement strict shellfish bans, and coordinate rapid response effort. Okay, so for example, in Aklan, okay, when there was a massive red tide in Aklan, okay, uh, three towns declared state of calamity. Okay, also in Bataan, when there was a red tide way back in 2021, okay, so state of calamity was also declared. And also some barangay in Malampayasan, Palawan, when there was a red tide, they are actually pleading the fisher folks, okay, shellfish farmers are pleading the LGU to declare state of calamity. So red tide is not just an ordinary environmental problem, okay? It can affect livelihood. It can take away human lives. Okay, throughout the history, the Philippines has experienced several major hub events. So here you can see on the left side, okay, that's the Philippines, and the red dots, these are the areas where uh, there was poisoning cases and deaths due to red tide, or technically toxic algal blooms. Okay, for example, in Manila Bay, somewhere in Leyte, you can see there several poisoning cases. For example, in Manila Bay, way back in the 80s and early 90s, there was 888 cases and 44 deaths. And also in Samar Leyte, in the Visayas area, okay, there are 279 cases, poisoning cases, and 21 deaths recorded. So way back in 1983, actually the first recorded red tide confirmed happened in Samar Leyte area. Okay, and the most recent happened in Bohol in 2015. Okay, there was also red tide that happened in Bohol. So most of these red tide events actually happened in the Visayas area, also in Mindanao. Okay, and one of the major culprits, okay, suspect of hubs in the Philippines is the dinoflagellate known as Pyrrhodium bahamense. Okay, because this dinoflagellate produces potent neurotoxin that can accumulate in shellfish, okay, leading to paralytic shellfish poisoning, especially when people consume contaminated shellfish. That is why the Bureau of Fisheries and Aquatic Resources has been mandated by the government to issue, okay, to issue bi-monthly shellfish ban, yan, warning, especially in this red tide affected area, to ban, okay, there's what we call a shellfish ban, to ban the selling, the harvest, and the transport of shellfish affected by red tide. Okay? Ayan. So to combat the dangers posed by red tides, okay, the government has realized that this type of phenomenon is very important. So over the years, our government, okay, have funded several research programs and projects dealing with harmful algal bloom, understanding their bloom mechanism, starting from the basic research, okay, basic research, studying the physiology, biology of the causative organism, especially the Pyrrhodium bahamense. Okay, and luckily I have been involved doing research as part of these different projects. So I started as a research assistant in UPMSI way back in 2009. So this is a very big hub program on the ecology and oceanography of harmful algal blooms in the Philippines. So I have been involved as a research assistant, then another project, senior research assistant, then senior research associate, and currently I also been involved in another large project funded by the Commission on Higher Education, or the CHED, funded by CHED Lakas, okay, CHED Lakas project, as project staff. And currently, in our small laboratory, we are implementing, okay, three externally funded project, mostly by DOST and UCC ERICA, studying harmful algal blooms, okay, mostly on the taxonomy and physiology. Okay, so since I have been involved with this different hub project, I realized that the harmful algal bloom problem is too complex, too big for one scientist or a small group of scientists to solve. That is why this type of problem, okay, needs multidisciplinary and collaborative approach among different group of scientists. You have there from biologists, taxonomists, oceanographers, okay, geologists, chemists and biochemists, ecologists, social scientists, okay. It's also important to have social scientists and artists, okay, to convey the information to the fisher folks, fisheries technician, to LGU, and policymakers so that they can understand the science. So that's very important, science communication. Okay, because sometimes scientists are too technical. Okay, that's why we need the help of social scientists and artists. Okay, artists sometimes they are helpful, especially when they are promoting this information, disseminating this to, especially sa mga bata. Okay, and uh, through the years, I have been working as a taxonomist, okay, contributing data to this different group of scientists, okay, counting, analyzing, quantifying the causative organism. Okay, therefore, why does taxonomy matter in this large, complex phenomenon known as HABs? Okay, so based on my experience working with harmful algal bloom for several years, okay, for one, the problem is too big. So earlier, I just emphasized one organism. Okay, organism, of course, because this is the major positive organism in the Philippines. Okay, this has been killing people, killing business. Okay, but there are other organisms that are actually causing problem. Okay, in the fisheries aquaculture. Okay, so it is the job of the taxonomies to really identify them accurately, because sometimes we need to be sure of the suspect, because not all microalgae are toxic. Okay, so there are some species naman of microalgae that are non-toxic. So therefore, doing taxonomic work actually allows us to distinguish toxic and non-toxic species. So as here are the different marine harmful algae. So this is based on the IOC UNESCO. There are a total of 199 species. So you have here different group of microalgae. You have here dinoflagellates, cyanobacteria, and others. Rapidophytes. So this one has been discussed by Dr. Wyman Lam. So he is actually a specialist on rapidophyte taxonomy. Okay, but there are other species that are causing problems. Okay, that's 104. I think the number now has increased. Okay, and this microalgae can cause several types of poisoning. So earlier I have emphasized the paralytic shellfish poisoning caused by pyridinium in the Philippines. But there are other species that can actually cause paralytic shellfish poisoning, or PSP, aside from pyridinium, and that is Alexandrium. And not to mention the other types of poisoning caused by this microalgae. You have their amnesic shellfish poisoning caused by Pseudonitskia, diuretic shellfish poisoning caused by dinophysis, and of course, the fish kill and shellfish mortality is caused by this dinoflagellate. Okay, so it is our task to really provide accurate identification, okay, of this different causative organism, because we cannot manage what we do not know. Okay, we cannot mitigate what we do not know. So basically, taxonomy is a foundational science. Okay, when doing harmful algal bloom research. So for example, here is the image of Pyrodinium bahamense. On their left is that's Pyrodinium bahamense, the number one hub organism in the Philippines. But if you observe under the microscope water samples collected from red tide affected areas, you will also observe this dinoflagellate, Goniodoma polyhedricum. Okay, to the untrained eye, okay, this looks similar. Okay, but there are actually major differences in terms of morphology. For example, Pyrodinium, they always form cell chains, group of chains yan. They also have antapical spines, yung mga spines niyan diyan sa baba. Okay. So also the number of plates differs. So another organism, and it's very helpful, especially when you're doing taxonomy, is to include molecular identification. Okay, so here is our paper published in Philippine Science Letters. So we have provided the molecular data. Okay, molecular data of pyrodinium occurring in Samar, Zambales, Palawan. Okay, so that is actually very useful when other taxonomies doing molecular identification to identify their organism, especially when they blast it in the GenBank. So another organism that is actually very important for us taxonomists to distinguish is the Alexandrium. So not all species in the genus Alexandrium are toxic. Okay, these are actually Alexandrium minutum, and Alexandrium tamutum. So the species on the left, that's toxic dinoflagellate, the toxic Alexandrium. Okay, and the one on the right is actually non-toxic Alexandrium. So it's our task to really identify these Alexandrium species accurately. So we study their morphology. So these species are actually covered with thecal plates. We call these thecal plates. It looks like a samurai armor connected. So the shape, the number is very important to us. Okay, when accurately identifying these species. And of course, we usually integrate molecular identification. This is another paper published in the Philippine Journal of Systematic Biology. So I have characterized Alexandrium tamutum, okay, and provided the sequences, okay, 28S and IPS sequences of Alexandrium tamutum. For other researchers, okay, to compare, especially when they obtain their own sequence, they can just blast it and check it in the GenBank. So another species that looks alike is Gymnodium catenatum and Gymnodium impudicum. So this is actually another toxic, highly toxic dinoflagellate. Okay, that looks similar with Gymnodium impudicum. So again, we characterize their morphology. Okay, especially the hypocone and all these intricate details. But of course, very important nowadays, we also provide molecular data. So this is another contribution. We have published the morphology and the sequences of Gymnodium catenatum and Gymnodium impudicum in the Philippines. So again, especially when we're trying to identify, this is very important, combination of morphology and molecular. Ayan. And in addition to distinguishing the toxic and non-toxic, so taxonomy is also important because there's what we call as emerging threats. Okay, these are species that has never been studied before, never been reported before, but because we report their occurrence in our coastal waters, okay, it's very important now to consider these species as emerging threats. For example, in our laboratory, we have been focusing on Carignaceae. So these are actually, so earlier, Dr. Wyman Lam discussed about Carignaceae in Japan, causing major fisheries, fish kill, okay, fish mortalities. So here, we have actually identified the presence of Carenia mikimotoi, Takayama acrotropa, Carlodinium australe, Carlodinium zoanum, Carlodinium balantinum. So most of these species actually has never been reported before in some coastal waters in the Philippines. Okay, so some species of Chatonella. So before, in the paper we published with Dr. Wyman Lam, only Chatonella subsalsa was present in the Philippine waters. But recently, in our laboratory, we have been identifying the presence of Chatonella marina, and another one is Chatonella malayana. Okay, that's another fish-killing dinoflagellates. And other dinoflagellates such as Margaphyllidinium polycricoides and Diatom pseudonitskia species. Okay, earlier I mentioned about pseudonitskia causing amnesic shellfish poisoning. So this is a news clipping published by ABS-CBN. Okay, presence of diatoms that can cause sickness, memory loss, confirmed in Luzon. Okay, this is a news that has been hyped in the social media. Okay, so they said that the microalgae, specifically diatoms that can cause memory loss, has been confirmed in Central Luzon. So actually, this is a work of 8 taxonomies. Although the data is still not enough to really confirm that this species, because as again, not all Pseudonitskia species can produce toxins. Okay, that's why it's very important to provide accurate identification. Number 3, another important contribution of taxonomies in harmful algal bloom research is the description of new species. Okay, So unfortunately, not so many scientists are specializing on taxonomy of microalgae in the Philippines, but actually, this is a very good niche to work on because there are a lot of undescribed species in our coastal waters. So even in our lab today, we have a lot of undescribed species. Okay, some of them died already. Okay, back to the environment. But this is an example of the new species I have described uh, from Manila Bay. So we have, of course, when you are trying to describe a new species, you need to establish that it is morphologically and molecularly different from existing species. So we have thoroughly, critically characterized the morphology of this Carnodinium azanzae. Okay, Carnodinium is a Carinaceae. So this is a harmful genus of dinoflagellate. Okay. This one, we published this in the Journal of Phycology. So another species that we have described is the Heterocopsa philippinensis. So Heterocopsa is a genus of dinoflagellate that actually includes harmful species, shellfish, killing species, dinoflagellate that can cause shellfish mortalities. Ayan, so I have described another one, Heterocopsa, so I named it after our country, Heterocopsa philippinensis, okay, together with Dr. Wyman Lam. And recently, we have collected samples in Ubando, Bulacan, an area where there's always a recurring fish kill. Okay, so here we have collected and isolated cultured small dinoflagellates, including a new species, Scripshella lawsonensis. Okay, this species actually has been isolated in a fish killing area, although we are still doing continued research exploring its effect on marine vertebrates, fishes, okay, to really assess its impact, okay. So I just like to promote the book that we have come up with, working with harmful algal bloom research for many years. Okay, this is a user-friendly, student-friendly guide on harmful algal bloom identification, sampling, okay, sampling, identification of marine dinoflagellates commonly encountered in the Philippines. Okay, so that's actually first edition, so but we need to update this. So with a lot of data that we are having, so maybe we can come up with next edition. And we welcome collaboration. So if you have water samples, okay, we can actually collaborate and contribute to the next edition of this book. So this one is actually available in UP. They are being sold in UP Diliman. Okay, so what are the future directions in hub taxonomies? Okay, of course, just like any other technology, there are a lot of advancements. Okay, so there are actually other tools that we can use for quicker and more efficient identification. Of course, what we are doing is the hard way because we start with culturing, okay, isolating, culturing, characterizing, but these information are very important and also can lay the foundation for future advancements. Okay, because we provide the sequences in the GenBank and that can be used, for example, for qPCR detection, development of LAMP detection kit, nano-based kits, and other these new technologies on molecular identification. Of course, the problem is too big. Okay, I have been mentoring a lot of baby taxonomies, young taxonomies in our lab because as you know, we are an archipelagic country. The problem is too big. Okay, a lot of coastlines, a lot of embayments, and we cannot solve the problem alone. That's why we need to mentor, train new generation of taxonomists, biologists, who are interested, okay, interested in doing this type of work. Okay, and of course, there's an opportunity to discover new species. Okay, and I also want to promote that our laboratory is open for collaboration. So you can just search our Facebook page, okay, CLSU Algal Diversity and Bioresources Laboratory, or you can email us at CLSU at adb at icloud.com. Okay. So to summarize, okay, harmful algal blooms or hubs is actually a major threat to public health, marine ecosystem, and fishing industry. And taxonomy is actually very important because it plays a crucial role in distinguishing toxic from non-toxic species, understanding emerging threats, okay, that has never been identified before or reported before, and identifying new species and describing new species. Therefore, taxonomy, with all these fields, okay, taxonomy is the foundational field for multidisciplinary hub research involving other fields of specialization, oceanographers, ecologists, fisheries technicians, chemists, and policymakers, social scientists, okay? Okay, with that, I'd like to thank my collaborators from UP, from ASCOT, also from University of Tokyo, our funding agency, okay, Department of Science and Technology, CHED, okay, also my collaborators from the Bureau of Fisheries and Aquatic Resources, okay, and my research assistants and students who are also actually the new generation of ecologists and hub taxonomists. With that, thank you very much. I'm ready to answer your questions.
All right. Thank you so much, Dr. Benico, for such an enlightening and thought-provoking presentation on naming the bloom, why taxonomy matters in harmful algal bloom research. Your insight into the critical role taxonomy plays in understanding and mitigating harmful algal blooms have shed light on a vital yet often overlooked aspect of the environmental science. So we are now ready to address questions from our participants. Please allow me to read each question for you, Doc. So for our first question, are there toxic harmful algae in freshwater ecosystem similar to the red tide?
Yes. In fact, I have an ongoing project. There's what we call as toxic cyanobacteria. Okay? So the group of microalgae that actually mainly responsible for harmful algal bloom events in freshwater ecosystem are cyanobacteria. Okay, some cyanobacteria also produces potent toxins, okay, hepatotoxic, neurotoxic, okay, that can actually contaminate freshwater ecosystem. So ang major concern dito ay yung sa mga drinking water, mga watershed. In fact, in La Mesa Dam, blooms of microcystis, a species of cyanobacteria, is a big concern. Okay? So it's not limited to marine. In fact, I have a project funded by the UC Cerca. We are actually determining the distribution of toxic cyanobacteria in Pantabangan Dam, Pantabangan Lake. Okay, determining the toxic cyanobacteria and also the gene responsible for the production of microcystin. Okay.
All right, thank you, sir. Next question is, why harmful algal blooms are more concentrated in Visayas?
Okay. So for one, there are a lot of islands. Islands in Visayas compared to Luzon, compared to Mindanao. Okay, the more islands you have, the more coastline, the more embayments, the more chances of having this type of blooms. Okay, and another one, there actually most of the it's in the Visayas where the red tide of pyrodinium started. Okay, technically the well-documented report started in Summer Leyte. Okay, so way back in 1983, the red tide toxic algal bloom of pyrodinium started in Summer Leyte. Okay, and it spread. Okay, I did not discuss in detail the life cycle, okay, the type of toxins they produce, but definitely this type of dinoflagellate can actually become cyst. Yan, parang buto. Parang kung sa tanim pa, parang buto, and they can easily spread. Yan. So in our internal cysts, especially in Visayas, there's one and actually may dalawang hypothesis yan nga, transfer ng cyst sa mga from one area to another, or another hypothesis is there actually for many years they have been there because of anthropogenic influences, high nutrient, growing population, the ecosystem, the balance of ecosystem has been disturbed. That's why dumami sila, especially itong mga toxic species. Because itong mga toxic brinoply, they are part of the ecosystem. They are there. They are normal, okay? Normally distribute occurrence. Pero yun nga, dahil climate change, okay? Increasing population, eutrophication. Yan, eutrophication. Increasing nutrient inputs, aquaculture, yan, overfeeding, yan, nagko-contribute din yan sa harmful algal blooms. Just to feed our growing population. Okay?
All right. Thank you so much, Doc, for that. For our next question, what measures can be implemented to mitigate the negative impacts of red tide harmful algal blooms on marine life and coastal economies?
Okay. We cannot fight nature. Okay? When they bloom, what we can do actually, in the grand scheme of things, that is why the problem is too big. What we can do is to manage. That's why our government has mandated the Bureau of Fisheries and Aquatic Resources to issue bi-monthly, two times, bi-monthly shellfish ban to this area. And of course, there is actually mga personnel who are responsible for safeguarding, monitoring, okay, monitoring our coastal waters. So dalawang way, either they obtain shellfish, okay, and extract the toxins, quantify the toxins. Actually, yung Bureau of Fisheries and Aquatic Resources, they are issuing shellfish ban based on the toxins detected in the shellfish. Okay? Kasi itong mga toxins, ito yung mga produce ng mga microalgae. Okay, once the toxin level goes beyond the regulatory limit, okay, I think that's 50 microgram per shellfish, 100 gram shellfish of meat, they will issue shellfish ban, meaning it is not allowed to buy, to sell, to transport. Well, kasi baka sabihin, dito may red tide, dito sa isang area na to may red tide. Dalhin ko kaya sa Laguna, dalhin ko kaya sa Pangasinan. Kaya wala namang red tide doon. So bawal din yan. Okay, hinuhuli yan. Okay? Kasi ang red tide is not an ordinary issue. It can kill. It can kill human lives. Okay? Not only it can kill business, it can kill people. That's the most important thing to do. That's why before, okay, actually doing the good job. We have been training before monitoring personnel to really accurately identify these toxic species. Okay? And another one, okay, dyan yung sa pyrodinium. Meron din pang other species, yung gaya ng diniscuss ko, may mga emerging threats. Ayan. So, yung mga ibang species naman that can cause fish kill, may mga technologies naman na dinevelop. Okay? For example, there's what we call as, when there is a bloom of coclodinium, there's what we call as clay mitigation technology. So, ang ginagawa, for example, sa Korea, kapag may red tide ng coclodinium, that's another species, nag-spray sila ng clay. Okay, nag-spray sila para mawala at mag-attach yung coclodinium at mag-flocculate. Yan, bumaba. So, this actually reduces the impact of fish kill, of coclodinium resulting to fish kill. Meron ding mga biological control. Yan. Algicidal bacteria. Actually, marami naman. But sometimes when the bloom is too big, okay, the coverage is too wide, it needs cooperation na talaga. Not only of 1 to 2 scientists, okay, it needs policymakers, it needs fisheries technician, it needs social scientists, artists, okay, because it's the problem is too complex and too big.
Yeah, okay. So next question, sir. Given the increasing occurrence of red tides and their devastating impacts on aquaculture production, is it possible to design a multidisciplinary approach integrating omics, techniques, ecological modeling, and remote sensing be utilized to predict and mitigate red tide events? In aquaculture zones, develop early warning systems for farmers or identify and implement sustainable strategies to minimize economic losses and ensure food security.
Okay, thank you for that Miss Universe na question. Okay, so basically, as I've discussed earlier, our government has realized this problem. Okay, there are a lot of researches na that has been done. Actually, may mga models na na-develop targeting itong Pyrodinium bahamense. May mga models na na-develop for early prediction, okay? Early prediction and forecasting. Ayan, utilizing models, remote sensing data. So actually, nagawa na yan specifically for Pyrodinium bahamense. Okay, kasi ito yung #1 killer sa Pilipinas. Okay, of course, application as many technology that we can apply, we need to apply it. Okay, sometimes we just need the support, funding support. Ayan. So ngayon sa Pyrodinium naman, we have progress. Actually, we are in Southeast Asia, we are one of the most admired group of scientists doing harmful algae. Kasi sa Pyrodinium research, marami ng mga technology na na-develop, detection kits, models, okay, that can actually predict. At this one can be applied sa different aquaculture areas, shellfish farms, okay?
All right, thank you
Thank
so
you.
much, Doc. Moving po for our next question. Does the classification of whether harmful microalgae or not depend on the water quality parameters, just like the pH, nitrate, temperature, dissolved oxygen, etc., as well as if it is either fresh or salt water?
Okay, so definitely it matters yung mga water quality parameters because depending on the species, okay, there are some species that will actually bloom in highly eutrophic water, high phosphate, high nitrate, okay? There are some species then that will actually bloom in a clean water. Mapapansin niyo na lang, for example, El Nido. El Nido is a very beautiful place, tourist spot. Yan. Pero nagkaroon ng red tide ng Coclodinium. So it's actually more on advection, okay, advection, okay. There's actually an existing bloom from one area at nadadala siya ng current system, okay, tides from one area to another. Okay, of course, different species has different requirement. Ayan.
Okay, we have here a question regarding if your laboratory, sir, accept interns from other universities daw po.
Yes, of course. We need to work through the process. So please send us an email, okay? And also our university, to our department, will coordinate with you. Of course, marami din kasing pumapasok. So kung may slot, so naka-schedule yan. So in fact, we're accepting interns from Mamasu, from TSU before, okay? And also a lot of high school students who are the future, okay?
Thank you.
All right, thank you so much, Doc. Another question related po sa ating presentation. Is it possible species of alga and cyanobacteria cause HABs in a specific area?
Sorry, what's the question? Multiple species. Yes, it's very possible, okay? Some of them actually can co-occur, what we call as co-occurrence. Dinoflagellate and cyanobacteria, this is very rare, but two dinoflagellate species, yan, can occur. But dinoflagellate and cyanobacteria, okay, to the best of my knowledge, parang wala pang ganun. Okay? Pero diatoms and dinoflagellate, ayan, gaya ng diniscuss ko kanina, maraming group of microalgae to, dinoflagellates, haptopites, diatoms, okay, they can actually occur several co-occurrence, magkakaroon ng co-occurrence of species bloom.
Okay, thank you, sir. I think that was our last question.
Thank you.
Tara na, bumaba na tayo
Okay, next part is an info session. Let us all watch this video and be oriented about the GSPS. Oh, sorry. Yeah. Okay, Sir Gary, please allow us to extend our appreciation. Okay, so allow me to read the context of this certificate. Republic of the Philippines, JSPS Alumni Association of the Philippines, in collaboration with Central Luzon State University, Science City of Muñoz, Nueva Ecija, Certificate of Appreciation is awarded to Dr. Gary A. Benico for his valuable contribution in providing his technical expertise during the JAAP webinar series of 2025 with the theme Exploring the Diversity and Wonders of Marine Environment, organized by the GSPS Alumni Association of the Philippines in collaboration with Central Luzon State University on March 5, 2025. Given this 5th day of March 2025 via Zoom at Central Luzon State University, Science City of Muñoz, Nueva Ecija, Philippines. Signed, Dr. Evaristo A. Abella, President of Central Luzon State University, Dr. Renato Reyes, President of Aurora State College of Technology, Dr. Karl Marx A. Quiazon, President of GSPS Alumni Association of the Philippines, and Dr. Yoshio Otani, Director of GSPS Bangkok, Thailand office. Sir, thank you so much po.
Thank you very much.
All right, next part is our info session. Let us all watch this video and be oriented about the GSPS scholarship grants and fellowship opportunities. Please feel free to use the raise hand if you have any questions about these grants.
Hello, thank you for the opportunity to speak with you today. I would like to introduce JSPS and our international programs. JSPS is one of the funding agencies of Japan, established in 1932. Annual budget is about $2.0 billion. We support curiosity-driven research activities. It means, in principle, we don't decide any specific theme in advance. Hello, thank you for the opportunity to speak.
Okay, once again, if you have any questions about this grants or opportunities about the GSPS
I would like to introduce
scholarship,
JSPS and our international programs.
please type in your questions in the chat box or use the raise hand feature. Okay, apologies po. We will be back right after addressing this technical difficulty po. Please stay tuned po. Okay, reminding everyone to please accomplish the attendance form, the evaluation form or links provided in our chat box for you to acquire your certificates.
Hello, thank you for the opportunity to speak with you today. I would like to introduce JSPS and our international programs. JSPS is one of the funding agencies of Japan, established in 1932. Annual budget is about $2.0 billion US. We support curiosity-driven research activities. It means, in principle, we don't decide any specific theme in advance, and we leave it to each individual researcher's curiosity. We call it in other words a bottom-up approach, so we cover all fields of not only the natural sciences but also of the humanities and social sciences. To realize this policy, we invite applications through open calls, and we work hard to ensure fairness in its review process through peer review. This slide shows our international programs. Our international programs focus on mobilizing researchers and building broad and strong international collaborative networks. These programs have 5 pillars, as shown here, and today I will introduce the programs shown in red. These pie charts show the number of researchers exchanged through our international programs. In fiscal year 2019, more than 3,000 foreign researchers were invited to Japan, including 43 from Philippines, and more than 4,600 Japanese researchers were sent abroad, including 69 to Philippines, through our international programs. Unfortunately, due to COVID-19, the numbers were significantly reduced in fiscal year 2020 and 2021. But in fiscal year 2022, it has been recovering to the level approaching as before the pandemic. JSPS has been carrying out bilateral programs in the form of joint research projects and seminars based on MOU for more than 50 years. This slide shows our basic scheme for carrying out bilateral collaboration. Under this scheme, JSPS and its counterpart funding agencies jointly select projects and provide matching funds. 2024, we founded 10 new research projects out of 93 applications. This program lasts for 1 to 3 years, so we have 31 ongoing projects in this fiscal year. JSPS offers another bilateral program without MOU, called Open Partnership Joint Projects and Seminars. In this scheme, JSPS supports researchers in Japan, and the counterpart researchers are not required but expected to secure their own matching funds from any funding agencies. JSPS offers another bilateral program without MOU, called Open Partnership Joint Projects and Seminars. In this scheme, JSPS supports researchers in Japan, and the counterpart researchers are not required but expected to secure their own matching funds from any funding agencies. This slide shows our international program for multilateral research collaboration, the Core-to-Core program. Application of this project requires the participation of 2 or more partner countries. It means Japan, 1 ASEAN country, and at least 1 other country. This slide shows ongoing multilateral collaboration programs between ASEAN countries and Japan in fiscal year 2024. 21 programs are now operated under this Core-to-Core program in ASEAN. This slide shows our fellowship programs for inviting foreign researchers to Japan. JSPS has a variety of invitation fellowship programs for overseas researchers, including PhD students and mid-career researchers. As you can see, despite of COVID-19 pandemic, postdoctoral fellows from Philippines are invited every year to Japan through this program. This slide shows our fellowship programs for inviting foreign researchers to Japan. JSPS has a variety of invitation fellowship programs for overseas researchers, including PhD students and mid-career researchers. As you can see, despite of COVID-19 pandemic, postdoctoral fellows from Philippines are invited every year to Japan through this program. This slide shows our fellowship programs for inviting foreign researchers to Japan. JSPS has a variety of invitation fellowship programs for overseas researchers, including PhD students and mid-career researchers. As you can see, despite of COVID-19 pandemic, postdoctoral fellows from Philippines are invited every year to Japan through this program. JSPS supports the activities of alumni associations of former fellows who have participated in JSPS fellowship programs. We offer Bridge Fellowship Program by which overseas researchers who participated in a JSPS fellowship program in Japan and joined an alumni association after returning their home country are provided opportunities to revisit Japan. We now have more than 8,000 members in 20 alumni associations. JSPS supports the activities of alumni associations of former fellows who have participated in JSPS fellowship programs. We offer Bridge Fellowship Program by which overseas researchers who participated in a JSPS fellowship program in Japan and joined an alumni association after returning their home country are provided opportunities to revisit Japan. We now have more than 8,000 members in 20 alumni associations. JSPS supports the activities of alumni associations of former fellows who have participated in JSPS fellowship programs. We offer Bridge Fellowship Program by which overseas researchers who participated in a JSPS fellowship program in Japan and joined an alumni association after returning their home country are provided opportunities to revisit Japan. We now have more than 8,000 members in 20 alumni associations. Hello, thank you.
Okay, to officially close our program, we have the President of GSPS Alumni Association of the Philippines, Dr. Karl Marx Sequiazon. Sir.
Okay, so distinguished speakers, esteemed guests, colleagues, and participants, as we come to the conclusion of this enlightening webinar series on exploring the diversity and wonders of marine environment, I would like to extend my deepest gratitude to our esteemed speakers for sharing their invaluable expertise and insights. To Dr. Tarman, she has highlighted the immense potential of marine biotechnology and its application, opening new avenues for innovation and sustainability in marine resources utilization. Dr. Loom has taken us through the fascinating yet concerning world of harmful radiophytes, particularly Chatonella, shedding light to its taxonomy and phylogeography in Southeast Asia. To Sir Marco de Guzman, who has provided us with a closer look at the seagrass ecosystem along the coast of Dilasag, Aurora, emphasizing the ecological significance and the need for conservation. Lastly, Dr. Ganico Benico has underscored the crucial role of taxonomy in harmful algal bloom research, reminding us why accurate species identification is fundamental in understanding and mitigating marine ecosystem threat. So this webinar series hosted by JSPS Bangkok, JSPS Alumni Association of the Philippines, Central Luzon State University, and Aurora State College of Technology has served as a platform for meaningful discussions, knowledge exchange, and collaboration. It is through gathering like this that we strengthen our commitment to marine research and conservation, ensuring that we continue to explore, protect, and sustainably utilize our vast and diverse marine environment. Once again, thank you to our organizers, speakers, and all of you who have actively participated. May this be the start or the continuation of more collaborative efforts towards advancing marine science. As stated by John F. Kennedy, we are tied to the ocean. And when we go back to the sea, whether it is to sail or watch, we are going back from whence we came. Safe travel to those joining from different locations, and we look forward to seeing you in the future engagement. Maraming salamat at mabuhay tayong lahat.
Okay, and that concludes our webinar on behalf of the GSPS Alumni Association of the Philippines and Central Luzon State University. We thank all of our distinguished speakers, guests, and participants for being part of this meaningful discussion. We hope that today's lectures have provided valuable insights and contributions to your research and ongoing projects. Our passion for safeguarding and preserving marine biodiversity and ecosystems has undoubtedly been sparked. Thank you to everyone who participated. We look forward to welcoming you again at our future webinars. Wishing everyone a great day ahead. Thank